Let me check if at all I am audible as well as visible to all of you. Ayyo. Am I? Yes, I am audible. Hello, good evening. Good evening, Manoj. How are you, dear? I hope you're doing great. So we'll just wait for some time, and I request every one of you who's probably watching this video to make sure that you share this video link with all of your friends who are there, so that everyone else can also join, and together we learn in a better manner, and it is going to be a fun class if all of you are there. So listen to me carefully. Your FA examination is going to come really soon, probably in the next week itself. It is going to start for almost a lot of you. And here in this particular video, I have just one aim, and that is to make sure everyone who watches this video until the end is going to be pretty sure that they are going to get the best marks out there. Without even a second thought, they are going to believe that. The reason why they're going to believe that is because every one of you will get a chance to ask your doubt. Every one of you will see that each and every question, even MCQs, are going to be covered in the best manner possible. Yes, can I have a quick little hi? Hello. Hello, Anmol Singh. Yes, I definitely. Pehchana kya? Obviously, yes. Oh my God. So your seniors are also here, my dear children. Okay, cool. So in this particular session, I have a very important thing to tell you before I go ahead, and that is, I am planning to cover sexual reproduction in flowering plants, and apart from that, I am planning to cover human reproduction. All MCQs, all important questions which are going to come from this chapter is going to be covered right over here. What about reproductive health, and what about the next thing? Reproductive health, and the next thing, which is the principles of inheritance and variation chapter, is going to be done in the next part of this particular session, which is going to be happening day after tomorrow. Okay? Hello Daya and Manoj, it's so sweet to see you as well, dear. Okay, so are we ready? Everyone watching this video is super ready. We are going to have full swing energy, and if you're ready, just give me a quick thumbs up. If we are having energy, just give a thumbs up. We'll go ahead with the MCQ questions. I hope I'll score out of out after completing this session. Manoj, definitely for 2 chapters, definitely, because I am planning to cover 2 chapters very nicely over here. So for these 2 chapters, complete marks. In the next section, next 2 chapters, complete marks. Okay, Manoj gave me a thumbs up. I request every one of you to be energetic, okay? Chalo, here goes the first question that we have. Children, you can give me the answer like this. 1, A, B, C, D. Okay, whatever your answer is, just give me the question number and then give me the options like this. Select the correct sequence of anther wall layers. Over here, you have got all the options. I request every one of you to go on, keep guessing it. Epidermis, middle layer, endothecium, tapetum. Epidermis, endothecium, middle layer, tapetum. Epidermis, tapetum, middle layer, endothecium, and tapetum, middle layer, epidermis, endothecium. What do you think is going to be the correct answer for this particular question? Come on. Everyone can guess, and always remember. So Manoj says the answer for this question is B. Okay, what about others? Dear children, do not shy away. I always tell this thing. I'm not here to judge you. I'll never do that. Okay, I'm just a good friend of yours, and I want to make sure that you get great marks. For that, obviously, there are be trial and error methods. You're going to give me wrong answers. I'm going to correct you. As simple as that. But you have to try. So Manoj says the answer for this question is B. Okay, let me log B. Is it B? Epidermis, then endothecium, then middle layer, then tapetum. Okay, the answer for this question is actually B. Okay, and if you are someone, here goes the answer, if you're someone who forgets it, mostly, I'll tell you a way to remember it, a trick to remember it. So over here, you have the anther wall. Okay, this is how the anther looks. There are 2 lobes. This is going to be the first lobe. This is going to be the second lobe. Each lobe has 2 theca. This is the first theca and this is the second theca. Now the outermost layer that we have over here is going to be epidermis. You call this as epidermis, just like what we saw over here. Second layer that we have just within it is going to be known as, by the way, this diagram is important, okay? It is going to be known as endothecium, just like what we discussed over here. Now inside that you have middle layer, but before I go ahead with middle layer, I'm making something over here. I'm going to make something over here. Just give me a quick second. Yes, this particular thing is made up of so many cells. What do you call this thing as? This thing is known as sporogenous tissue. It is known as sporogenous tissue, and this tissue is made up of multiple cells. What cells? They are going to be the microspore mother cells, as in the ones that is going to form the pollen grains later on. So all these cells who are going to form pollen grains later on are right over here. They're hidden over here. And to nourish them, you have a wall layer around it. You have a layer around it which you call as what? Tapetum. Ma'am, you didn't talk about middle layer. Arey Baba, middle layer is actually in the middle. So this particular layer that you have over here in the middle is going to be known as what? It is going to be known as the middle layer. Jim Broom Bra, will it work? So sometimes it does not work. Yes. So this layer is going to be known as middle layer. And as I said, for those of you who are getting confused, I'll tell you a way to remember it. Ears come first, then comes mouth, and then comes throat, right? Ear, mouth, and throat. So you can remember EMT. Epidermis, endothecium, okay? Epidermis, endothecium, you have two of them, epidermis, endothecium, middle layer, and then tapetum. EMT. So I used to remember it like this. It's pretty easy for me to remember. So you go to doctors, ENT doctors, right? ENT doctors, ear, nose, throat doctors. With that, I came up with this idea. I hope this idea is going to help you get marks as well. So please remember, if this question is super clear to you, if you're okay with it, can you give me a thumbs up? And if you like this particular trick out there, then also please give me a thumbs up. Clear? Can we go ahead with the next question? Children, if you do not give me thumbs up, I will not understand whether you are understanding things or not. Yes, perfect. Thank you, Manoj. Next question that we have over here is select the correct sequence of events in microsporogenesis. So here goes microsporogenesis, and you have to tell me what is the correct sequence of microsporogenesis. How does the whole process happen? Sporogenous tissue, then microspore mother cell, then microspore tetrad, then microspores. Here you have microspores, then microspore mother cell, microspore tetrad, sporogenous tissue. You have multiple options out there. You have to give me the correct one. Manoj said the answer for this question is A. I really want all the other chutku little children, machas, also to give me the answer. Baba, can you try once? I'm telling you honestly, with utmost amount of honesty, I'm telling you, I'm not going to judge you. Please give me the answer. Okay. So I think slowly you will become friends with me. So I'll go with the first option that Manoj has given me. He says that this is the answer for the question. Microsporogenesis. Is this the correct answer? Yes. Actually, A is going to be the correct answer. And for those of you who have no idea why is it A, let me tell you. So I spoke about the sporogenous tissue which had so many cells. Okay, and I told that basically these cells are going to make pollen grains later on. How do you think the whole process happens? I am going to talk about microsporogenesis. So for those of you who are not clear with this particular concept, listen to me carefully. Over here, each cell, Baba, each cell is diploid in nature. It has 2n number of chromosome, as in the normal number of chromosome a cell has. Okay, and I'll call this as microspore mother cell. I'll call this as the microspore mother cell. What do I mean by that? I mean that they are the mothers of microspores. But ma'am, you said that they are going to form pollen grains. How can we be the mother of the microspores? So Baba, first they form the microspores, then these microspores themselves, they transform into what? Pollen grains. So that is why I can call them as pollen mother cell. I can call them as what? Microspore mother cell. There are so many of them inside this. You call this whole thing as sporogenous tissue. So the first thing that we would be having is going to be sporogenous tissue. Yes ma'am, done. Okay, then over here I told microspore mother cell is there. This microspore mother cell which is there, it is going to start undergoing a division which is known as meiosis. Why meiosis? Baba, whenever you are going to form a gamete or something that is related to a gamete, you have to make sure the number of chromosome become half. Why? Because always remember, a gamete carrying half number of chromosome, another gamete carrying half number of chromosome, when they come together, when it carries half and half, and when they come together, they form 2n. And 2n is the normal number of chromosome that we have in all the cells. So microspore mother cell, which is a normal cell, is going to undergo meiosis. And meiosis happens in 2 stages. This is the first stage of meiosis, where you get 2 cells. where you are going to get 2 cells. And in the first stage itself, we are going to see a very, very important thing, that is the diploid number of chromosome, that is 2n number of chromosome, is going to become half. It is going to become half. It becomes n. Now what happens? Again there is meiosis, because as I said, meiosis is going to happen in 2 stages, Baba. So again it is going to divide. It is also going to divide. So ma'am, this was n. Will it reduce to again half of it? The answer is no, it is going to remain the same. Meiosis 2 is just like mitosis. The cell number increases, the number of chromosome remains the same. So reduction happens over here. Anyways, at the end, what do you get? At the end, you are going to get microspores. And you know what about these microspores? These microspores are already very much together with each other. So all of them, I'll say, they form a tetrad. They are going to form tetrad. What do you mean by that? They are saying that I cannot stay alone. I have always been with my siblings. I am going to stay together. So this group of all the 4 microspores are known as microspore tetrad. When they are formed like that. Later on, this is all happening inside the anther. Inside the anther, they will be facing dehydration, as in the level of water is going to get reduced. At that time, all of them, when suppose you are there in a closed room, you are staying with your family and friends, but one day the water, food and everything ends. All of them are going to be like, oh my God, I cannot take it over, and you all will be running here and there. Similarly over here also, when the anther dehydrates, all the microspores from the tetrads, they are going to move away from each other, and finally we say that they are individual microspores. They are what? Individual microspores. So Baba, we had sporogenous tissue. Out of that, all the cells are made up of microspore mother cell. All of them are going to divide like this. All of them are going to form microspore tetrad, and the microspore tetrad, from them, all of them are going to divide from themselves and form individual microspores, and these individual microspores are later going to form your pollen grain. Is the pollen grain the gamete? No. Pollen grain, inside the pollen grain, you would be having the gamete. Pollen grain is not the gamete, it has the gamete, which is why I'll call this as gametophyte. The one that is going to be having the gamete is known as gametophyte. So male gametophyte is nothing but the pollen grain, and this is microsporogenesis. So I have a question over here. Deepak is asking, ma'am, how long will the class be? So the class will be until 8:30. Okay, it's correct 3-hour class. I'll try my maximum not to extend it. I'll try my maximum to cover as much as I can within this particular time itself. The 2 chapters that I have in my target, I will try to complete it such that it will be easy for you. You will feel that at the end of this class, you will feel that, okay, I feel good. Got it? Yes. Cool. So the answer for this question is going to be A. I hope this is clear. Guys, this question is paka clear. Can I go to the next one? Can I go to the next one? If yes, give me a thumbs up, please. If this is clear and you do not have any doubt out there, give me a thumbs up. We'll move on to the next question. Perfect. Thank you. Okay. Which of the following has proved helpful in preserving pollen as a fossil? Okay, which one is that thing that had helped pollen to remain as a fossil? You have pollen kit, you have cellulosic intine, you have oil content, you have sporopollen. What do you think is the answer for this question? What is the reason such that I can preserve the pollen grains as fossils? Manoj says D is the answer. Is D is the answer? What about others? Deepak, gonna try, dear? Let's check. And everyone who feels that this class is useful, make sure you share it with your friends. Okay, so I'll go with D because I do not see any other answers coming up right now. So sporopollen is what Manoj is saying. Is that the answer? Yes. Listen to me very carefully. We're going to talk about the pollen grain. By the way, there are small things given over here. These are notes for you, and I'll tell you where you can find it. Anyways, so over here you have the pollen grain. Okay. Inside the pollen grain, you have 2 cells. Actually, I told you there was a microspore. If you remember, we had just formed the microspore. The microspore, which is having haploid number of chromosome, is going to undergo mitosis. Is going to undergo mitosis, as in cell division, cell dividing into 2 cells. Over here, both the cells are together here itself, forming the pollen grain. So when I say this cell divided into 2, both the cells are here itself. I'm going to tell you how. So one cell is there, this big cell that you see over here. You call this as the vegetative cell. Vegetative cell. How do I remember? V for very big. Okay, V for very big. So there's a very big cell over here. The shape of the nucleus is important. This has come in one question. This has come in one type, 1 year for the question paper, and it is what? Irregularly shaped nucleus. It is irregularly shaped nucleus that you have. It does not have a proper shape. Now this is the cytoplasm of the cell. You know what happens? Within the cytoplasm of this particular cell lies a small cell out there. Lies a small cell out there with elongated nucleus. This cell which is there is known as generative cell. What do you call this as, Baba? Generative cell. Ma'am, why do you call it generative cell? And what is the shape of the nucleus? It is going to be elongated. It is going to be elongated nucleus. Now, ma'am, coming back to the question, why do you call it generative? Generative means it is going to generate something. What could it be generating? Any guesses, guys? What could it be generating? If I say this is generating something, what is the next thing that this particular pollen grain is going to generate? Any idea? Any idea? Generative cell will turn into male gametes. Perfect, macha. Yes, this generative cell which is there, Baba, it is going to form gametes. How many gametes? Can you imagine? 2 gametes. 2 gametes. So 2 male gametes. Ayyo, where is it? 2 male gametes are going to be formed over here. Okay, and this is a very important question. Always remember, you have a vegetative cell. Inside the cytoplasm of it, you get a generative cell. The generative cell later on, not now, later on is going to undergo mitosis. So first mitosis, you got these 2 cells. Second mitosis happens, that's when you are going to get 2 male gametes. Cool. Now this is a pollen grain. It will be having its own plasma membrane and everything. It has the plasma membrane. But apart from it, it has something that is super important. What is that? There is a continuous layer like this. There is going to be a continuous layer like this. What do you call this layer as? You call this layer as, any idea? You call this layer as intine. It is inside something. That means outside something else is also there. Yes, I am coming to that. Before that, this is intine. Do you know intine is made up of what, Baba? No, Manoj, it's not exine. It is going to be intine. It's going to be intine. We are going to talk about exine really soon. Any idea what is intine made up of? Anyone who is watching this video have a wild guess out there that intine could be made up of this? Come on, children. We will not be friends if at all you do not talk to me, no. So you have to talk to me. Otherwise it is going to be a boring lecture. Okay. So then I am believing that no one is ready to tell. No one probably knows it, so I only have to tell. So I'll tell you. Intine is made up of, again, yes, Manoj, cellulose and pectin. So I have a pretty easy way to remember it. Priyanka Chopra. Okay, it's Priyanka Chopra. You might have heard about her. Priyanka Chopra. P for pectin. C for cellulose. Intine is made up of Priyanka Chopra, that is pectin and cellulose. There is an external layer outside this, and this is very important. Listen to me carefully. The external layer which is outside over here, just a second. It is going to be like this. So, ma'am, why are you making like this? There are pores that you are making over here, no? Yes, there are small pores, very small pores that are there. By the way, this particular layer that I have is known as exine. And exine is made up of the most important thing on this world, and that is going to be sporopollenin. That is going to be sporopollenin. You know what? Suppose I have acid over here. Okay, I have acid over here. I put it on the pollen grain. Still it will survive. Why? Because pollen grain is surrounded with exine. Exine is made up of sporopollen, and sporopollen is the most toughest material out there. Even if I put acid, if I put alkali, if I put whatever I want, still it will be able to survive because of this guy which is there. And this is exactly why the pollen grains can be remained as fossils, because for fossils, obviously fossil is the dead remains that is there, which can withstand anything, which can stay out for many years. So it can also do so. The reason is this. Okay, and as Manoj was saying, over here you see some spores. It's not spores, sorry, pores. Okay, what do you call these pores as? You call them as germ pores. What do you call this as? You call it as germ pores. Why do you think germ pore is there, Baba? Listen to me carefully. This is the male gamete that we have made. Later on, they are going to form the male gamete. These male gametes have to move out, no? They cannot always stay inside the pollen grain like this. So for that, they need a way. They need a way, and that way is this germ pore. Through the germ pore, they are going to make a tube-like structure. Who is going to make? Who is going to help? Vegetative cell, which is there. It is very big. Why do you think it's very big? First of all, to provide nourishment. Second reason is that it gives all its cytoplasm. It gives all its cytoplasm to form a tube-like structure, which is known as pollen tube. It is known as pollen tube. Exactly, dear. So pollen tube is being made, and through the pollen tube, the gametes which are going to be formed are going to move down. So that's why you need germ pores. That is why you're going to need germ pores. I hope this is clear. This is the male gametophyte. If at all you have been asked about the diagram of male gametophyte, Baba, you have to draw this diagram. Always remember, vegetative cell, very big cell, irregular nucleus. This guy, elongated nucleus. I know I'm taking time in MCQs, but trust me, you're going to require less time for 2 marker and 3 marker and 5 marker later on. Okay, this is the male gametophyte diagram. If it comes, makes the diagram of the pollen grain. Perfect. Can you give me a thumbs up? If it's clear, can you give me a thumbs up, everyone? Or if you have any doubt, please ask me right away. Don't think that I'll ask later on. Okay. So I got 1 thumbs up. And by the way, I want to tell one thing a very important thing to every one of you. So those of you looking for the notes, first of all, it will be given to you in the WhatsApp channel, definitely, for sure. Where can you find the WhatsApp channel? Over here. So suppose this is the live that is ongoing right now. Over here, you will be able to find the WhatsApp channel. If at all you just, Diksha KCET WhatsApp channel. You can just click on this link and you will be guided there. Apart from it, there is another very, very important thing, and that is this. So there is a all-in-one page. All-in-one page means everything that you're seeking forward for. The classes, suppose in YouTube, no, there are so many distractions, there are n number of videos out there. But you need only academic lectures. So you can find it over here. Okay, this is the link that is given in the description section. Over here, you have to go and click on buy now. Do not worry, it's for free. It's for free. You just have to fill in your mobile number and basically you would be able to access it. Yeah, you will be able to access it. And for you guys, you will not have this part, okay? You will not have Pariksha Vira, but you will have ACE KCET 2027. You just have to click over here. You will get to see biology, physics, mathematics, everything. So if I click on biology, all the videos that I have done for biology is going to be covered over here. And in the coming days, you are going to, you're not able to see it. Okay, now you're able to see it. So all the videos that I have done over here will be visible. And apart from it, okay, there will be assignments that is going to be given to you. The sample papers for your FA and all will be shared over here. So I'll request you join the WhatsApp channel, and in the WhatsApp channel also this link for the AIO page, all in one page, will be given. And apart from that, if you want to directly go and check out, you can go and check out. You just need to fill in your mobile number. I hope that is not a big of issue. Right, so this is one thing that I wanted to tell you guys. Okay, and I'll be closely reminding you this again and again, because a lot of you might be coming a bit late to join the class. That's why. Okay, so now going ahead with the next thing. The next question that is there, and it's going to be like this. Select the incorrect statement with respect to vegetative cell of the pollen grain. Come on. Select the incorrect statement with respect to vegetative cell of the pollen grain. Now you will be able to solve this question because we had just discussed about it, right? It's bigger than the generative cell. Vegetative cell, do you think it's bigger than the generative cell? That is the first option. Second option, it has abundant food reserve. Does it have? It has a large irregularly shaped nucleus. Is it? It floats in the cytoplasm of the generative cell. Is that true? So Manoj said the answer for this question is going to be D. Anyone has any other opinion? No, ma'am, D is not the correct answer. If you have, please let me know. Well, the answer for this question is going to be D itself. And by the way, for everyone who's thinking that where are these questions from, these questions are taken from the question bank. So most of the important questions from the question banks have been taken, bank that has given previous year have taken over here. So you need not to be worried about solving the question bank later on. Okay. So Baba, I told you this. It's actually correct what Manoj has said. Listen to me carefully. You have to find the incorrect statement about what vegetative cell. But we already know all the statements are correct except one. It is actually bigger than a generative cell. We saw the picture. Okay, we actually saw the picture. The next thing that we have over here is that it has abundant food reserve. Does it have abundant food reserve? Yes, that is going to give this food reserve. It is reserving all this thing for what? It is reserving all this thing for the papa guy out there. That is your generative cell, right? Now then after that, it has a large irregularly shaped nucleus. Do you think it has it? Yes. By the way, Mohammed Safan is also there saying that D is the answer. Perfect. Da. Okay. It has irregular nucleus, that is also correct. It floats in the cytoplasm of the generative cell. This is going to be wrong because it floats in the cytoplasm of the vegetative cell. Right, the generative cell floats in the cytoplasm of the vegetative cell. It's not the other way around. Got it? Okay, so the answer for this question is going to be D, as you guys said. Perfect. And I'm super proud of you. You're doing great. By the way, children, am I audible? There is no stuck and all in between, no? Because I feel there is some network issues, no, right? Okay, so I'll go ahead with the next question then. In that case, guys, give me a thumbs up if it's clear. A quick thumbs up if it's clear. I'm so sorry I'm making you do thumbs up, but I do not understand whether you're understanding or not apart from it. Okay, perfect. Let's go ahead then. Next question is, which one of the following statement is not true? You have to say which one is not true. First one, pollen grains of many species cause severe allergies. Is that true? Second thing, stored pollen in liquid nitrogen can be used in the crop breeding programs. Third one, tapeworm helps in dehiscence of anther. Fourth one, exine of pollen grain is made up of sporopollen. What do you think is the answer for this particular question? You have to find which one is not true. Don't find the one which is true because you will be confused. All of it is all of it is going to be true, right? So Manoj has already given me the answer. Is there anyone else who says that no, Manoj, there's some other answer, or is there anyone supporting and saying that yes, this is the answer? Mohammed Safan, want to try, dear? C is the answer for this question. Let's check it out. Okay, so pollen grain of many species cause severe allergies. That is true actually. A lot of them have allergies caused by the pollen grains, and the most important plant that usually causes allergy is known as parthenium. It's known as parthenium. You call it as carrot grass. You might have seen it. Green grass with basically white flowers, small flower sort of thing. Okay, so this is correct. Stored pollen in liquid nitrogen can be used in crop breeding programs. Is that true? Yes. Listen to me very carefully. The pollen grains which are there, if I put it in liquid nitrogen, you know what's the temperature of liquid nitrogen? The temperature of liquid nitrogen is minus 196 degrees Celsius. You do not have to maintain this temperature. Liquid nitrogen means the temperature is going to be like this. Liquid nitrogen means minus 196 degrees Celsius will be there. And you have to add more and more liquid nitrogen. That is the only thing that you have to do. If you keep it, it is going to be like that only. The pollen grains will be preserved, and basically later on, suppose I feel that this plant is going to die, and if I preserve the pollen grains like this, I can use these pollen grains later on for breeding the plants and for making plants similar to this, such that the plant does not get extinct. So this particular point is also correct. Now, tapeworm helps in dehiscence of anther. What do you mean by dehiscence of anther? Dehiscence means breakdown of anther. You need to listen to me very carefully. Tapeworm, which we discussed for the first time over here, if you remember, the innermost layer that is nourishing all the cells in the sporogenous tissue, that is the tapeworm. That is tapeworm. And tapeworm is the only guy that is there for nourishment. It's the only one which is there for nourishment. What about the three other layers which are there? The three other layers which are there, they are there for two reasons. One is for protection. Oh, I am going to protect all the cells which are over here. That is one thing. That is protection. And the next one which is going to be there, apart from protection, they also do one more thing. That is, they help in dehiscence of anther, as in breakdown of anther, such that all these pollen grains which are there are getting released. So I'll say protection and dehiscence. Protection and dehiscence, both of them is handled by these three guys out there. Tapeworm is for nourishment, and I'll tell you one unique feature about tapeworm which you need to remember. Okay, listen to me very carefully. Tapeworm is a layer which is made up of cells. What type of cells? So these cells, now suppose this is a cell. This cell had a nucleus. Now the nucleus divided into two, thinking that the cell also will be dividing into two. The nucleus divides into two. This is known as karyokinesis, nuclear division. Later on, it expects that the cell will also divide into two. That is known as cytokinesis. But you know what happens? The cell does not divide into two. You have one cell with two nucleus. Baba, what's the use of this? Why do you have a condition like this? Listen to me carefully. The only reason why we have this is because while you do this, okay, it has the content of 2 cells. The cytoplasm is really bulged now. It has enough cytoplasm, and we need the cytoplasm for nourishment of all these cells which are inside. That is why actually we have lots of dense cytoplasm and 2 nucleus. We wanted more cytoplasmic content, that is why we allowed the karyokinesis to happen, but cytokinesis does not take place. So always remember one feature about this guy, that is it will be having dense cytoplasm. It will be having dense cytoplasm, and apart from it, it will be binucleate in condition. That means it will be having 2 nucleus. It will be having 2 nucleus. You can expect these type of questions also coming up there. Don't think that exact questions similar to what we're talking about will come abruptly like that. It is not going to happen. There are going to be some changes. Please note that. Okay, so tape term helps in dehiscence of panther is a wrong option, and as Manoj said, this is the correct answer. Exine of pollen grain is made up of sporopollen. Yes, it is made up of sporopollen, and here you have got the answer for the question as well. Cool, works. Can we go ahead with the next question? If yes, can you give me a thumbs up? And all my dear children, please listen to me carefully. Make sure you send this particular video link with all of your friends out there. With all of your friends out there, very important. Okay? Just a second. Cool. Perfect. The next question that was there is question number 6. The functional megaspore in an angiosperm develops into an what? So functional megaspore in an angiosperm develops into what? Endosperm, embryo sac, embryo, ovule. What do you think is the answer for this particular question? Functional megaspore. We are talking about megasporogenesis. Okay? Megasporogenesis. If at all you remember the steps of it, you will be able to give the answer for this as well. Manoj says the answer for this question is B. Manoj, you have been super correct all this while, da. Yes, you are saying that the answer is B. Is the answer B? Let's check it out right now. So listen to me very carefully. Again, I'm bringing back things over here. Inside the ovule, okay, inside the ovule, that is known as megasporangium. Okay, there also the picture that we made of the anther. Inside the anther we spoke about multiple wall layers. Basically we're talking about microsporangium there, where you have sporogenous tissue which is going to make the pollen grains and all of it. Similarly over here, ovule is your megasporangium, and inside the ovule you have only one megaspore mother cell. You have only one megaspore mother cell. This megaspore mother cell is going to divide. Meiosis, again it becomes NNN. This is meiosis 1. Again meiosis happens, you get 2 cells. Here also you are going to get 2 cells. The number of chromosome is going to remain the same. This is meiosis 2. Now what is the thing that happened? You are basically expecting 4 megaspores to form. Right, you are expecting that 4 megaspores are going to be formed, but I'll tell you a twist in the story. 3 of them are going to say tata bye bye, I'm getting degenerated. Only 1 is left behind, and that is going to take care of all the things. So this one guy, this one megaspore that stays there and says that, hey macha, I am going to take care of it. I'll make the gametes and all. I'll make the female gametophyte first. Okay, just like how you have pollen grains there. No, microspores form the pollen grains. Similarly, I am going to form into the female gametophyte right over here. This type of development, by the way, is known as monosporic development. What do you call it? Monosporic. What do you mean by monosporic? One spore develops into the entire female gamete out there. Only one, rest all of them degenerates. What is a female gametophyte known as? It is known as the embryo sac. It is known as the embryo sac, and that is the question that has been asked to you guys. That is, Baba, the functional megaspore in an angiosperms develop into what? You know the answer. It is going to be the embryo sac. So the answer for this question is going to be embryo sac. Okay, and you are correct, Manoj. Got it? Any confusion that you have, any doubt that you have, you want to ask me, ma'am, how does this transition happen? What is the changes that is going to happen? Anything, please ask me right now. If it's clear, give me a thumbs up. If everything is clear, super clear, just give me a thumbs up. Perfect. Thank you. The next question that we have is question number 7. It goes like this. In angiosperms, microsporogenesis and megasporogenesis involves meiosis, occur in the ovule, occur in the anther, form gametes without further division. What do you think is the answer for this question? In angiosperms, microsporogenesis and megasporogenesis has what? Has what? And here are the options that is given to you. Come on. Manoj says the answer for this question is going to be A. Okay, what about others? Any guesses, children? Any guesses that you have for this question? A involves meiosis. So megasporogenesis and microsporogenesis we saw in both the cases. There was a megaspore mother cell, here also microspore mother cell. Both of them were definitely undergoing meiosis for sure. So this particular statement is not wrong. But we have to see whether there is other statement which is more correct or not. So megasporogenesis and microsporogenesis occur in the ovule. Arey Baba, no. Only mega will occur over here, micro will occur in the anther. No. So obviously we cannot say yes. Why? Because you have been asked about both of them together. Now, form gametes without further division. Arey they are going to definitely divide and then only they can form the gametes. Without division, how can someone form the gametes? So the answer for this question is going to be what, Baba? It is going to be A. It is going to be A, and we have got the answer. We have got the answer. A involved meiosis. And Manoj, you were correct. Ayo, Manoj said D. Manoj said Dia. Dia. It will be A, no? Karthik, hello dear. I hope you're doing great. The answer is going to be A, Manoj. Any doubt, children? Everyone who is watching this silently and not thinking, I am not going to comment. I'm just going to watch this video without commenting. Do you think it's okay that you're able to understand it? If yes, can you just give me a thumbs up? Guys, I'm waiting for you. If you have understood, can you give me a thumbs up? Ma'am, I got confused and sent. It's totally fine, Masha. You got it right now. No, that's what I want. Okay, I hope that's clear. And now we go ahead with the next question out there. And it goes like this. Okay. Function of filiform apparatus is 2. What is the function of filiform apparatus? What does it do? I actually said A itself. Yeah, I saw that. That's what. I saw, and then again when I turned back, it was D. So I was like, Anyways, function of filiform apparatus is to what? Recognize the suitable pollen at the stigma, stimulate division of the generative cell, produce the nectar, guide the entry of pollen tube. What do you think is the answer for this? Manoj now says it's D. Arey, wah, wah, wah. Yes, it is actually D. It is actually D. Now, for those of you who are like, what is filiform apparatus? What is ma'am talking about? Listen to me very carefully. I'm just going to take you back to history. Okay, a little bit of history over here. You come on, much work. Okay. So over here, I told you that this particular megaspore which was there, it formed the embryo sac. That is the female gametophyte. Mahantesh is also there. Hello dear. Mahantesh also gave me the answer as D. Now the structure of embryo sac is something that I am going to talk about right now, because this question is about it. Function of filiform apparatus is 2. In the embryo sac itself, you have the filiform apparatus. If at all you do not know its location, probably you might not be able to tell its answer also. So for everyone who is confused, listen to me carefully. By the way, this is a picture that Claude had made in, but anyway, I'll make a picture for you guys. So this is your embryo sac. Inside the embryo sac, over here, you have 3 cells. They are always together, and they are haploid cells. You know what's their name? Their name is antipodal cells. Antipodal cells. They are dancing on apadipoda, okay? Antipodal cells at the top, they're just chumma sitting there. Okay, then over here, where are you? You have 2 cells which are again haploid. What do you call these 2 cells as? These cells are known as synergids. What do you call them? Synergids. Again, they are also haploid. Okay. Now there is our VIP member over here. This VIP member is known as egg cell or the gamete. This is your gamete. The number of chromosome is going to remain the same. It is going to be having haploid number of chromosome, obviously. Now, oh my God, Malli Serigiri is also there. Hi dear. Malli Serigiri is also saying it's D. Now that's the Josh. Okay. Now finally over here, we have one very big cell. This big cell that you see over here, where is it? Come on. Jim Brumbra work, yes. This big cell that is there, you have a name for it. You call this as a central cell. Okay, the central cell which is there, it has two nuclei, not one but two. All this papa guys were having only one nucleus. The central cell, which is very big cell, says that I am very big, I need two nucleus. Okay, take two nucleus. One nucleus over here will have haploid number of chromosome. This also will be haploid number of chromosome. So can I say n plus n, this cell will be diploid in nature. Only cell which is going to be diploid in nature. Ma'am, what are you talking about? We were talking about something else. No, over here, I am coming to that. Okay, so yes, as Manoj said, these nuclei which are there, they are known as polar nuclei. Okay, now you have been asked about what is the function of filiform apparatus. Ma'am, you didn't speak about it. Filiform apparatus which is there is right over here, macha. On the synergids over here, you have finger-like projections like this. They are known as filiform apparatus. What do they do? So you know what? Pollen tube which is there. You know, I told you pollen grain, they have germ pores through which the pollen tube is going to come. The pollen tube is going to carry two male gametes and is going to come over here. Just give me a second. This has some issues. Ayyo, where are we? 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 I hope I do not show you any answers. Yes. Okay. So they are carrying two male gametes and they are coming over here. When they are carrying gametes and coming over here, papa pollen grains, they do not know. Sorry, the male gametes do not know where to go. They are like, okay, should I go there? Should I go here? That's when the filiform apparatus say that, hey, macha, I am going to help you enter into the embryo sac. I am going to guide you. So that is the role of filiform apparatus. It says that, macha, you come through this place. Here is your egg cell. You want to meet with it? No? Go meet. No problem. Okay, that is the thing. Filiform apparatus has a role of guiding the entry of pollen tube. Please remember it. It does not have any of the other function. Recognize the suitable pollen at the stigma? No. Stimulate division of generative cell? No. Produce nectar? No. Only thing is this. And I have a question for all of you who is watching this video. Don't think that I'm just gonna solve MCQ and I'm gonna leave you like that. I'll be asking questions to you. You tell me, embryo sac which is there, it is inside the ovule, right? It is there inside the ovule. On which side of the ovule do you have the antipodal cells, and on which side do you have the synergid and the egg cell? There are two sides, chelazal end and there is micropylar end. So I'm asking you, in the micropylar end, what do you have? I'm asking you a question. In the micropylar end, what do you have? Do you have the antipodal cells who are doing apodipode, or do you have the synergids and the egg cell out there? In the micropylar end. Come on. Antipodals. Ullas says antipodals are there in the micropylar end. Dan-ta-dan. Ullas, by the way, hello dear. Okay, egg apparatus. Okay, Manoj says egg apparatus. Whom should I give the score? So listen to me carefully. I am really Mahantesh says synergids. Three different way of answering it. Anyway, so listen to me. The answer for this question is micropylar end. Micropyle is what? A small opening. Opening where will you need? Over here, such that the pollen tube can come in. It's the easiest way to remember. Do not ratify, I'm telling you. Use the common sense which is there. You have an opening. Through that opening only pollen tube can come. No? So over here in the egg apparatus, this is the egg apparatus, synergids, plus the egg is known as egg apparatus. Over here you are going to have that here. So micropylar end is right over here. Please write it and remember it. And the chelazal end will be having the antipodal cells. Chelazal end is going to have the antipodal cells. Okay, fine. So Ullas got it, and I'm really glad you guys are trying. I'm super proud of you. Okay, that's what I want. Give me wrong answer, give me right answer. That's how we communicate. That's how we become friends, right? So everyone, Ullas, Manoj, Mahantesh, and yes, everyone. Malli Sirigari, where are you, dear? Come on, participate. So I hope this part is clear, dear children. This is known as embryo sac, and you have all of it. Always remember, all the cells over here are haploid, except this guy central cell, which is diploid. If it's clear, can you quickly give me a thumbs up? We'll go on to the next question. If it's clear, give me a thumbs up. We'll move on to the next question. And by the way, if no one told you, let me tell you, you guys are amazing. Okay, and you are giving your best over here. That's why you're watching this video. Not so many people are watching this video. Why? Because they think that they probably are going to do it later on, or probably they have their own favorite teachers. That could also be a reason. But I'm telling you, you are ahead. You're already ahead because you have made up the mind and you're watching this video. You're giving your best. right? So good work. Perfect. Let's go ahead with the next question. That is there, question number 9. From the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. Now, what are you being asked about? You're talking about true about the female gametophyte. What is a female gametophyte, Baba? We all know the female gametophyte is what? Come on. Female gametophyte is going to be what? Then we'll talk about the options. Female gametophyte is the other name for pollen grain, something else, ovum, sorry, not ovum, what do you call ovule? It is going to be what? Female gametophyte is nothing but the embryo sac that we just spoke about right now. So the first thing that is given over here, Manoj has already given the answer. Okay, embryo sac. Mahantesh also said embryo sac. Perfect. It is 8 nucleate and 7 celled at maturity. That is the first option. 8 nucleate and 7 celled. Let's check it out. Okay. 7 celled, let's check that out first. 1, 2, 3, 4, 5, 6, 7. 7 cells are there. What about the nucleus? 1, 2, 3, 4, 5, 6, 7, 8. Oh my God, it is correct, ma'am. 8 nucleus. 1 extra nucleus is there because one big guy was very bad. This guy was selfish and said that I want 2 nucleus because I am very big. Okay, remember like that. So nucleus is extra and that is why 8 nucleate and 7 cell at maturity. So this is correct statement. Statement number 2 says it is free nuclear during development. What do you mean by that? It is free nuclear during development. Do you have any idea about that? We'll keep that on hold right now. We'll keep it on hold and we'll move ahead with the next option. Okay. It is situated inside the integument but outside the nucleus. Again, we did not know about it until now. It has an egg apparatus situated at the chalazoid. We really know about this. It does not have egg apparatus in the chalazoid end. Egg apparatus is there in the micropylar end. So this is going to be wrong. This is wrong. This is correct. So I have A option. Wherever I have A option, I am going to tag them. Okay, B and C and everything I'm just going to cut. Wherever I have A, I'll just put it on hold. I'll just put it on hold. Either this can be the answer or this can be the answer. Now, here 2 terms are given over here. Okay. Ma'am, right after commenting, you're not able to see it. Oh, is it? So, I think it could be a slow... okay, it could be a slow network issue or something like that. Okay, anyways. So, we have 2 things that we are not sure about right now. So, I'm going to talk about it right now over here. I told you, if you remember, that a megaspore is there and that megaspore is going to form the embryo sac. Okay, that is how it is going to work. How do you think that usually happens? So here is the story of that. Suppose this is the megaspore that we had, and this megaspore had a nucleus that was having n number of chromosomes. Okay, it had n number of chromosomes. Now we have to form an embryo sac with so many cells. We started off with just 1 cell. We have to make so many cells. So what are we going to do? This is very important. So listen to me very carefully. The nucleus over here is going to divide itself. If there was 1 nucleus, it is going to divide into 2. The number of chromosomes remains the same. That means we are talking about a division which you call as mitosis. Ayo, come on, work, macha. Okay, I'll call this division as what? Mitosis, macha. I'll call this as mitosis because mitosis is the only division where the number of chromosomes remains the same. Similarly, again over here, the nucleus divides into 2. This nucleus also divides into 2. Okay, the number of chromosomes again remains the same itself. It does not change. It remains the same. It remains N. So this is what, ma'am, this is mitosis. Then here also you have mitosis. What mitosis? The second round of mitosis. Okay, second round of mitosis. So I'll write, ayo, come on, work, macha. Oh, patience, patience. Okay, this is going to be mitosis, second round. Okay, again this is going to divide. Again this nucleus divides into 2. Again this nucleus divides into 2. Again this nucleus divides into 2. This also divides into 2. So I'll say there is third round of mitosis that is happening up over here. Third round of mitosis. Okay, the number of chromosomes, will it remain the same? Yes, it's mitosis, which is why it is going to remain the same itself. These 3 mitosis which is happening, you have to note one thing. The nucleus is dividing. Does it have any cell wall formation around it right now? No, there is no cell wall formation that is happening right now. When they are dividing, they are not having cell walls being formed around it. This type of division, where you do not have immediate cell wall formation, is known as free nuclear division. It's known as free nuclear division. Is that happening over here? Yes. Once all of the nucleus is being formed, later on the cell wall is going to surround. Okay, the cell wall and a little bit of cytoplasm is going to surround all of them. Now all of them, one will be having 2 nucleus. This is going to be the central cell. Okay, rest of the arrangement, it goes like this. 3 cells from here go at the top and arrange over there. You call them as antipodals. You call them as antipodals. 2 of them say, Macha, I'm going over here. I am going to be known as synergid. 1 of them again come over here. This is going to be known as the X cell. Okay, and then remains the very big cell which has 2 nuclei. That is how things are going to work. This is how things are going to be working. Cool. I hope it helps you. This is known as free nuclear division, and later on you will be getting all of it. So that was the question that was asked over here. It is free nuclear during development. The answer is yes, it is free nuclear. And how many mitosis do you require, Baba, to form the female gametophyte? You require 3 mitosis. In the case of male gametophyte, if you remember, the microspore divided by mitosis, you got 2 cells, vegetative and generative. This again divides, the generative cell again undergoes mitosis, you get 2 gametes. So 2 rounds of mitosis happens there. Here 3 rounds of mitosis are going to happen. That's how you get the female gametophyte. Okay, now there is one more part of it, that is, it is situated inside the integument but outside the nucellus. What do you mean by that? Now for that you need to go ahead and talk about the structure of the megasporangium, or basically the entire ovary and things like that. Okay, ma'am, your voice is little bit cracking. Oh my god, what all is happening? Hello. Let me check. Little bit cracking. Oh my god, what all is happening? Hello. Can you check, Mahantesh? Because here when I hear it's not cracking, dear. Is it some issue with or probably it could be my hair doing the job. Okay, now I put my hair back. Should be fine. Okay, now guys, I am going to go ahead. I know that almost this question has been solved. We have got A and B as the correct one. So probably the answer for this question is going to be C. Did someone give me the answer as C? I think yes. Manoj had already given me the answer as C already. Okay, Manoj had already given as C. Okay, now I'm coming back over here. Can I go ahead and talk about the structure of, the ovary, and inside that the ovule and everything? If yes, can you give me a thumbs up? Or if you want me to go ahead with the next question, I can. But you will not understand this, and probably you will not understand the further questions that are going to come. Sharath Kumar, ma'am, I have a big doubt. Please ask me, dear. Ma'am, when will math session? Math sessions are ongoing here. Tomorrow you have a math session in this channel. Okay, Mahantesh is okay with it. So probably we'll go ahead and talk about it. Just give me a second. So I need a place to draw. And this is the place. So listen to me very carefully, okay? Very, very carefully. This is a diagram of what? This is basically, I think, a 3-marker question that could be coming up to you. If you know this, it will be really, really good. Ma'am, next question. Okay, so basically since Manoj is saying next question, I'll come and talk about this. Listen, I am just solving a question right over here. You can think like that. Draw a neatly labeled diagram of anatropous ovule. This is a 3-marker question about which we are going to discuss now. So both of these things can be linked, and anyway we are talking about that over here. I hope that works. Okay, that is going to work for you. So in that case, we are solving that question, which is 3-marker. So it goes like this. You have to draw the diagram exactly like this. Listen to me very carefully. You have the ovary right over here. This is going to be the ovary right over here. Where is it? Come on, board. Yes. Here you have the ovule. Here what do you have? You have the ovule. So this is going to be the ovule. This is going to be the ovary. Now, this cavity that we have within the ovary, what do you call this as? You call this as locule. Okay, you call this as locule, or you can also call this as ovarian cavity. Guys, please draw your diagram like this. It is known as the ovarian cavity. Okay. Now, obviously it cannot be in air. The ovule cannot be in air. It has to be supported somehow. Okay, so in that case it will be like this. In that case it is going to be somewhere like this. This particular structure which is there, it's known as placenta. Oh my god, ma'am, some siren sound is coming when you talk. Siren sound. Okay. There is no siren that is happening up over here, da. There is no siren that is happening. I don't know. Let's see if it persists. Anyways, this is the placenta that you are having. The placenta is going to be attached. Okay, attached to what? The ovule, with the help of a structure which you call as funicle. It's a structure that helps in attachment of placenta to the ovule. The placenta is there for nourishment purpose. It is there for nourishment purpose. Okay, now right over here, the point of attachment that you have right over here, what do you call this as? This is known as hilum. This is known as hilum. Okay, the point of attachment is known as hilum. Now, one more very important thing is there, and with that we are almost going to cover up the entire picture. It goes like this. That is, the ovule which is there, it is inside the ovule, it says that I am feeling very cold, can you give me sweater? So, the ovule says that, yeah, please have 2 sweater, wear 2 sweaters. So, this is the first sweater that it has. You call this as integument. What do you call this as? Integument. So, this is going to be your, just a second. I don't know what is happening today. This is going to be your inner integument, and then there is one more integument out there which you call as outer integument. So similarly, you have 2 integuments. Now, if you see carefully, there is an area over here which is not filled with anything. Okay, this is not filled with anything. It's basically the ovule that is saying that I am not going to button it up. I want to show some style. So over here, I do not have anything. This pore that is there, Baba, it's known as micropyle. It is known as micropyle, and this end where you have the micropyle is known as the micropylar end. It is known as what? The micropylar end, and the opposite end of it, opposite side of it is going to be known as the chalazal end. It is going to be known as the chalazal end. Until this particular point, if it's clear, can you give me thumbs up? Until this whole thing is, if it's clear, can you give me a thumbs up? Quick thumbs up. And I hope the audio and all is audible, because you know what, when I am listening to myself, there is nothing like that. Okay, Manoj says thumbs up. Okay, fine, cool. Now we are coming back over here. This ovule, which you also call as megasporangium, has something within it. That is, first of all, it has a layer of cells right over here. What do you call these cells as? These cells together form a structure that is known as nucellus. It is forming a structure that is known as nucellus. It is there for nourishment purpose. Okay, it is there for nourishment purpose. Ma'am, what is it nourishing? Listen to me carefully. Here within it, you had the megaspore mother cell, which was also diploid in nature. Megaspore mother cell. Only one cell is there. In the case of microspores, they have so many microspore mother cell, but here only one guy is there. And in that one, when it divides, it forms 4 megaspore. 3 of them degenerate. Only one remains, and that one is going to form what? That one thing that remains over here is going to form your embryo sac. Is going to form your embryo sac, where you have the synergids, the egg cell, the antipodal, and the central cell right over here. Right? So now the question which has been asked over here says, or basically asks you one simple thing, and that thing is, where is the question? The question is like this. It is situated inside the integument but outside the nucellus. Is it correct or not? It is situated inside the integument. What? Embryo sac. Embryo sac is situated inside the integument but outside the nucellus. It will be wrong. It's situated inside the nucellus as well. Embryo sac is the innermost thing which is there. Okay, embryo sac is going to be the innermost thing which is there. Got it? Cool. Anatropous. Ullas, it is known as anatropous. Yes, it is just the opposite. I'm going to talk about that. So I hope that particular portion is also clear. Here the question says it is inside integument but outside nucellus. No. Nucellus, integument, everything is outside. Innermost thing that you have is the embryo sac. Now, the picture that is asked to you in your diagram is what? In the 3-marker question is anatropous ovule. If not anatropous ovule, if you are asked to make the megasporangium, make like this. This is the megasporangium and this is the structure around it. If you are asked the anatropous ovule, just note one small thing, and that is in anatropous ovule there is going to be some opposite thing that is going to happen. Here you will not have the funicle, here you will not have the hilum and everything. Here you do not have the hilum or the funicle or everything. Where does the placenta and everything go? It goes up here. So you will be having placenta over here, you would be having the funicle over here. This point of attachment that you have right over here is going to be known as the hilum. Just make it on this side. Chalazal end remains the same. Chalazal end remains the same. Micropylar end remains the same. Only difference that you're going to see is placenta, which was over here, moves this side. Funicle, which was over here, moves this side. Hilum, the point of attachment, moves this side. That's all. That's all. What do you call this type of ovule as? You call this ovule as anatropous ovule. You call this as anatropous ovule. And here is a picture of anatropous ovule for those of you who probably could be getting confused. Where is it? Where is it? Where is it? Yes. So see this particular picture very carefully, okay? What do you have? This is going to be your embryo sac. Outside the embryo sac, you have the nucellus. Outside the nucellus, what do you have? Outer and inner integuments. Here you have the micropyle. Opposite side of it, chalazal end. Near the micropylar end, you have the funicle. This is the funicle. The point of attachment is known as hilum. As simple as that. If you make the diagram like that, it is going to be done. Okay? Cool? Cool? Okay. Now I get it. So there is some issue with the... Is it fine now, children? Is it fine now, children? There is no PP, kiki and all, no? Is it fine? I'm so sorry, but I feel it was inside my pocket, that's why. If it's fine, can you give me a thumbs up, please? Okay, perfect. Ullas, I'm asking if they are orthotropous. Yes, the previous diagram that I made where you had the placenta and the funicle at the correct line, vertical line, that was orthotropous. Now the diagram that I had made over here, I had changed and made, right? This one is going to be anatropous. So the one that is straight is known as orthotropous, and the one that is like this is known as anatropous. Orthotropous term is not there in your NCET, that is why I do not go specify much about it. But yes, good one, Ullas. Okay, let's go ahead now. So I hope this question is clear. I hope the next concept is also clear. Now we go ahead with the next question. It goes like this. Identify the correct sequence of arrangement of cells in the mature embryo sac from the micropylar end. From the micropylar end, Baba, you have to be very careful answering this question, okay? You have been asked the correct sequence of arrangement of cells in the mature embryo sac from the micropylar end. Okay, not from the chalazal end, but from the micropylar end. Come on. Solve this question. Manoj says the answer for this question is A. Okay. What about R.S. Gaudaru, Ullas and Mahantesh, and there was someone else also. Tarun is there. Hi friends. Hello friends. Okay. Ullas says A. Okay. So two people are there saying the answer is A. What about others? Children, come on. Try Madi. And everyone who is watching this video, I hope you hit on the like and the subscribe button as well. Anyways, since you're going with A, let me also go with A. Let's think about it. Synergids. Yes, we know in the micropylar end, first thing that we have is synergids. Then we have the egg cell, correct. Then we have the central cell, correct. Then at the top we have the antipodal cells, that is the chalazal end, correct, Baba. So all of you, oh my God, R.S. Gaudaru is also correct, Tarun is also correct. Honey is there. Hello, Honey. I hope it's known as honey itself or honey. Okay, and Mahantesh also says A. Perfect. Now you guys made my day, okay? This is how it goes from micropylar to chalazal end, and all of you are correct, which is why I'm not explaining much. Cool. Works, right? Works, no? Can I go ahead with the next question? If yes, can you give me a thumbs up? Or if you have any doubt that you want to ask, you can ask. And I'm so happy. Thank you. Perfect. Okay. Let's go ahead with the next question. Which of the following is not a correct statement? You have been asked which is not a correct statement. Okay, placenta is located outside the ovarian cavity. Central cell develops into PEC after fertilization. What is PEC? It's basically primary endosperm cell. Okay. Orchid contain more than one ovule in an ovary. Egg apparatus consists of one egg cell and two synergid cells. So there are a few points that we already know. There are a few things that we do not know. But anyways, okay. Ulla says the answer for this question is A. Oh my God, perfect. Okay. Honey also says the answer for this question is going to be A. Manoj also says it is. Manoj says it is B. Okay. You have been asked which is not a correct statement. Okay. Done? No? Deal? No? I'm locking your answer and I'm going to give my answer. Listen to me. First statement. Placenta is located outside the ovarian cavity. Cannot be true, Baba. We just saw placenta is located where? We just saw it right now. Placenta is right over here. It's basically inside the ovarian cavity. In this particular cavity, it's there. So that statement goes off. Then what do we have? Central cell. Okay, we'll just pause this particular point for some time. We do not know about it as of now. Okay. Orchid contain more than one ovule in an ovary. What do you mean by that? Ovary. This is the ovary. Inside that you have the ovule. What does the ovary become later on? It becomes the fruit. What about the ovule? Ovule becomes the seed. So always understand, in plants which have multiple seed, that means you're talking about having multiple ovules. Watermelon, papaya, all of it is an example. Similarly, the example that you probably cannot recall could be orchid. Orchid, now you will not forget it. Orchid is O and it has so many ovules. Okay, it has so many ovules which transforms into seed. So this particular statement is also correct. Now, egg apparatus consists of one egg cell and two synergid cell. Okay, egg apparatus. Am I wrong somewhere? Okay, egg apparatus consists of one egg cell and two synergids. This particular statement is also correct. So you have been asked what is not correct. Arey bhai. Okay, so here this is not correct. Sorry. My brain is not braining. So it is located inside the ovarian cavity, but this is wrong because here it is mentioned outside the ovarian cavity. So this particular statement is going to be wrong. This particular statement is going to be wrong. A ma'am. I'm so sorry. Yes. I said it is inside, but I didn't see this particular thing that is outside. I'm so sorry. Okay, but anyway, so this particular statement is going to be wrong, but we are still not sure about this one. I mean, you all know this one, but still. Central cell develops into PEC after fertilization, primary endosperm cell. Is that true or false? Is that true or false? The answer is yes, it develops into primary endosperm cell. And right now I'm going to talk about that right over here. Listen to me carefully. I'm going with one thing that is known as double fertilization. I hope you guys do not mind. If you're okay with this, can I talk about double fertilization right over here? If you're okay, or I'll just move ahead. I'll just go ahead. So listen. Okay, you're okay with it. Thank you so much. So listen to me carefully. When I say that you have two male gametes coming up, okay, both the male gametes are going to be haploid. Two male gametes which are coming from that are going to be haploid, right, okay. Now one male gamete which is going to be there, it says that I want to meet with the egg cell. Egg cell is already haploid in nature, plus one male gamete comes up there. One male gamete comes up there, it is also haploid in nature. Haploid becomes what? It becomes diploid. What do you get at the end? You get the zygote at the end. You get the diploid zygote at the end. What do you call this process? This is known as syngamy. This is going to be known as what? Syngamy. So I'm writing it here itself. It's known as syngamy. Perfect. One fertilization done. The next fertilization for which you are doubtful about, ma'am, is the polar nuclei and things like that. Let me tell you. So the next male gamete that comes over here says that, okay, I do not have the egg cell now. Let me fuse with the central cell. Let me fuse with the central cell. The central cell has two nuclei. Central cell has two nuclei. Both of them are polar nuclei. And over here, you have got now the male gamete. Male gamete is also a cell. Always remember, male gamete which is there. Come on, work. Okay, male gamete which is going to be there is also a cell. Here this cell has haploid number of chromosome, this cell has diploid number of chromosome. These cells when they fuse, what do you get? You get the primary endosperm cell. You get the primary endosperm cell, which is going to be what? Diploid plus haploid. 2n plus n, it becomes 3n. Now, there is one more thing happening. This is a cell that is fusing. The nucleus is also fusing. No, the nucleus is also fusing. So over here, this is the nucleus of the male gamete. Just a second, okay? This is the nucleus of the male gamete. The two polar nuclei, which is diploid in nature, together, and over here this is haploid in nature. They also come together to form a nucleus, which is going to be triploid in nature. What do you call this nucleus as? This nucleus which is there, Baba, is known as... Ayyo, come on, pen, work, muddy. Okay, this nucleus is known as the primary endosperm nucleus. It is known as primary endosperm nucleus, which you also call as PEN. Pen. This is PEC, primary endosperm cell. Inside the cell, you have the primary endosperm nucleus, which is also going to be triploid. What do you call this fusion as? Anyone? Yes, Ullas. It is known as triple fusion. It is going to be known as triple fusion. Why? Because three nucleus are coming together. That is why you call it triple fusion. And since two fertilizations are happening, one over here syngamy, second over here triple fusion, I will call this as double fertilization. What do I call it? Double fertilization. Got it? So Manoj, when you're saying that, ma'am, it's polar nuclei, no, but so listen to me carefully over here. The question is what? Central cell develops into primary endosperm cell after fertilization. It develops into primary endosperm cell. No, we're not talking about primary endosperm nucleus, right? So that is why it would be correct. Anyway, it would be correct, but still. Yes. Hello, Sridhara. Hi, how are you? I hope you're doing great, dear. So everyone who is watching this, Hani, Sridhara, and Sampath is also there. Hello, Manoj and R.S. Gowda, Mahantesh, dear, all my dear children. Got it right? Characteristic feature of angiosperm, double fertilization. Exactly. Exactly. Double fertilization is a unique feature of flowering plants. Now, all of you who have got it, can you give me a thumbs up? That is how I work. Okay. When I see thumbs up, I understand. It's all cool. It's all cool, right? Understanding? No. Thumbs up. Ready? Perfect. Thank you so much. The next question that I have over here goes like this. I'm last year student, ma'am. Is it? Sridhara. So good. I hope everything good happened to you. Anyways, so, and I'm really glad you're here, by the way. Even in the absence of pollinating agents, the seed setting is assured in what? Cucurbita, papaya, commelina, or maize? It's an easy question, very, very easy question. Come on, try doing it. Ullas says the answer for this question is C. Okay, Manu says the answer is C. Everyone is saying C, C, C. Okay, okay. Next. Two people are saying C. What about others? Mahantesh also says the answer for this question is C. So it will be C. If you guys are saying it's C, then I have to lock C. The answer is actually C. So always remember, you're talking about what? You're talking about clistogamous flowers. So there are some type of plants which have something like this. Okay, they have open flowers where the male and the female reproductive system, they are all open, they're exposed like this. Okay, then they have something like this, the closed setup, where they have the male and the female together at one place, but it is closed. Now here, for here, pollination to happen, if it's very, very near, you do not need pollinating agents. The anther and the stigma, if it is very near, you do not need much pollinating agents. But here in this case, but sometimes you may need also, you may sometimes need, you may sometimes not need, depending on how the structure is. But in this case, anyway, you do not need any pollinating agents. Why? Because they are closed. They are closed. Where will the pollen grain go? It does not have anywhere to go. It will just come over here. So definitely, seed is going to be formed. Fertilization is going to be happening. When fertilization happens, I'll say that the ovule which was there, inside which the zygote is formed, I'll not call this as ovule anymore, I'll call this as a seed. So anyway, seed is going to be formed, seed setting is there for sure. This type of flowers are known as what? Clistogamous flowers. Okay, I'll call them as clistogamous flowers. CL stands for closed. You can remember like that. And these type of flowers that we have is known as chasmogamous flowers. They are known as chasmogamous flowers. Now, one thing that I want to tell you right over here, the examples of plants that have both these type of flowers are three. And I remember it in a very easy manner. Cow. But this time cow is with V, not W. Commelina, oxalis and viola. Commelina, oxalis and viola. So even if the option had something else similar to oxalis and viola, please make sure that is something that you go ahead with. Perfect. Cool. So the answer for this question is this. And by the way, I'll tell you an easy peasy fact, not funny fact, actually interesting fact. These flowers that you have made, these are flowers, okay, the clistogamous flowers. They're usually more towards the root region. Do you want to see actually how does it look? It looks almost like this. They look like this, and they are found either inside the soil or basically here. Why inside the soil? Because anyway, it does not matter if they are closed or not. Pollination is definitely, sorry, not pollination, yeah, pollination is definitely going to happen. Seed is definitely going to be formed. That is the reason. Got it? Clear? Can I go with the next question? If yes, can we give me a quick thumbs up? If I can go with the next question, please give me a quick thumbs up. And everyone who is watching this video, I have a sincere request to all of you. Make sure you share the video with all of your friends and ask them to join this live. It will be really great if so many children also come along. Okay, and my next class is going to be there on Friday, day after tomorrow. Okay, thank you so much for giving the thumbs up and let me know that you have understood it. Let's go ahead with the next question then. Which pollination brings genetically different types of pollen grains to the stigma? Which pollination brings genetically different types of pollen grains to the stigma? Geitonogamy, xenogamy, autogamy or clistogamy? Ullas says B is the answer. Manoj says B is the answer. Okay. What about others? Come on. B is the answer. Genetically different types of pollen grains you want. Okay. Okay, Mantesh also says B is the answer, which is why I'll log B, xenogamy. So you have geitonogamy, you have xenogamy, you have autogamy and clistogamy. You have to choose between them. The answer is going to be B, and I'll explain it to you right over here. So there's a very cute picture. This particular flower which is there, suppose it has the female and the male part right over here. Okay, it is going to do pollination like this. This is known as self-pollination. Within a single flower itself, the pollen grains are getting transferred. Okay, and this is known as autogamy. Self-pollination actually technically is known as autogamy. For it, you have multiple requirements. The requirement is that basically the flowers should be bisexual. That means it should be having male and female part both. Otherwise, how will it happen in the same flower itself? The second thing is that basically all this thing, that is anther, that is a stamen, as well as the pistil, should be almost of the same height and everything, such that easy pollination can happen. Even if without pollinators also, pollination can happen very easily. So structurally they should be almost together. The next one is that they should have a synchronization. That means when the pollen is released, the stigma which is going to receive it should be receptive. If it is not receptive, what is going to happen? The pollen grain comes and land and the stigma is like, you wait there for some time. But we do not want that to happen. We want the stigma to receive it, which is why you need synchronization as well. You need synchronization. Cool. Now here when self-pollination happens, do you think genetic diversity will be there? Do you think genetic diversity will be there? No, Baba. Why? Because pollen grain from the same plant, male, female gamete also from the same plant. How do you expect genetic diversity to happen? For that you need cross-pollination. No, this is self-pollination. Okay, ma'am. Then we go ahead with the next thing. That is geitonogamy. Ma'am, what if I have a flower over here, okay, which is having the female part, and here I have a flower which is having male part. Both these flowers are attached to the same plant. Two different flowers of the same plant. Here when pollination happens, what do you call this as? Since these two flowers are of the same plant, I'll call this as geitonogamy. This is known as geitonogamy. I cannot call it autogamy. I'll call it geitonogamy. Okay. So, ma'am, this is technically cross-pollination. Yes, definitely. This is going to be cross-pollination. Self-pollination happens within the same flower. So, this is going to be cross-pollination. Is it true cross-pollination? The answer is no. Why? Because again, the male part from the same plant, female part from the same plant. Genetically, are you going to get anything different? No. You will not get anything different over here. Genetically, you are not going to get anything different over here. Always remember that. This is very, very important. But this is cross-pollination, but I will not say it's true cross-pollination. Finally, you have got something like this that is known as xenogamy. Xenogamy is where you have two different plants. From here, suppose you have the female and here you have the male. From here to here, transition is happening, well, pollination is happening. Okay, is it true cross-pollination? Definitely, it is true cross-pollination. Genetically, do you think you will get different type of baby plants out there? Definitely, because two different plants are being involved. And that was the question exactly. The question went like this, that is, which one brings genetically different type of pollen grains? The answer is xenogamy. The best way to remember that it's a true cross-pollination is X. X means cross, and this is showing true cross-pollination. Geitonogamy is also cross-pollination, but not the true one. Got it? And by the way, every one of you who want all these notes, first of all, I request you to make sure you check out the AIO page. The link is given right up there. So join the free second PUC course is given right there. No, you just have to click on that link. You just have to go there and click on the buy now option. Click on the buy now option. Do not be worried, it does not have any money. It's free of cost. Okay, it is just free of cost. You have to just fill in your number and all. You will end up somewhere over here. You can go ahead and check each subjects, biology, physics, mathematics, everything is over there. Entire thing that is covered in the YouTube, you will be able to see that over here. You can play that video over here. Apart from it, soon we are going to upload some assignments also there. Okay, so this is all in one page. The link for that is given in the description section and also right over here in the chat session. Works? Okay, so I hope this question is super clear to all of you. If yes, can I go ahead with the next question? Can you give me a thumbs up? If yes, if I can go with the next question, can you give me a quick thumbs up? Okay, thank you so much, children. Dioecious plants are what? Dioecious plants are going to be what? Autogamous only, both autogamous and geitonogamy, both geitonogamy and xenogamy, xenogamy only. Can you tell me the answer for this? Okay, Ullah says the answer for this question is B. Manoj says the answer for this question is D. Okay, Mahantesh says it is B. So 2 votes for B, 1 votes for D. 1 vote for D. What about others? Anyone wants to try? What do you mean by dioecious plants, first of all? What do you mean by dioecious plants? Dioecious plants means their sexes are going to be separate. I'll say this is a plant that is going to be a female plant. And I'll say this is a plant that is going to be a male plant. Okay, in the female one, the flowers will be having only the female reproductive organ. In the male one, the flowers will be having only the male reproductive organ. So in this particular case, in this dioecious plant, autogamy is the only thing that happens. Do you think autogamy can ever happen? No, it does not have the male part. No, how will autogamy happen? So I'll cross that out. A is definitely not the answer. Both autogamy and geitonogamy. Geitonogamy again will not happen. Suppose over here, you have another flower. This is also containing female only. No, how can you have geitonogamy? That is also not possible. And autogamy was not all, as I already said, it was not possible. Both geitonogamy and xenogamy. Geitonogamy is not possible. Xenogamy is definitely possible, but half of it is wrong. That is why this is wrong. Last option that you have is xenogamous only, which is why the answer for this question is going to be D. And Manoj gave me the answer, but it's still okay. Don't retract your message. Listen, we have to face it. It's not an issue. No one is going to judge you. I'm telling you that. Who is going to judge you? Who is going to judge you? There's no one, literally no one. Now Manoj says, what is bisexual plant? Bisexual means they are known as monoecious plants. Just like how we have dioecious, right? So in dioecious, only one thing can happen, and that is xenogamy. Dioecious, xenogamy. Always remember that. Okay? I'll give you an example of that, and then I'll go ahead with the bisexual plant. Dioecious example is papaya. Papaya plant is dioecious. Okay, I'll show you a picture also if I have. No, I do not have. So it has the male plant, it has the female plant, like that. Now, since you are asking about what do you say, bisexual plant, I'll talk about the other thing, monoecious. Monoecious plants, they are like this. It can be 2 types of monoecious. Okay, one type of monoecious is where one flower is there, you have both the female and the male part here. This is a bisexual plant. Or there can be one more scenario of monoecious where one flower is female and the other flower is going to be male. The other flower is going to be male. This is also bisexual because it has both the sexes right over here. Okay, got it? Example for this one, you're going to see that in outbreeding devices. Outbreeding means what? You want cross-pollination to happen. If at all this is there, cross-pollination cannot happen. So there will be outbreeding devices. That means in the plant itself, they decide that I am not going to have flowers like this. Rather, I will be having something like this. This is somewhere going to help me. At least autogamy is going to be prevented. Geitonogamy still can happen, but at least autogamy is prevented. No, that is one thing. Then you have this thing. But anyway, but I hope you got it. Got it? If yes, can you give me thumbs up? We'll move on to the next question. Perfect. Thank you. So thank you all of you, and let's go ahead with the next question. And I hope everyone has the energy. My energy is going a little down, but with your energy it is going to resonate. Okay, so let's go. Read the two statements and choose the correct option. Two statements you read properly and then choose the correct option. First statement, in Viola, both autogamy and geitonogamy is prevented. Viola, both autogamy and geitonogamy is prevented. Second, geitonogamy is similar to autogamy since pollen grains come from the same plant. You have A, both statement 1 and 2 are correct. B, both statement 1 and 2 are incorrect. C, statement 1 is incorrect but statement 2 is correct. And then D, statement 1 is correct but statement 2 is incorrect. Manoj says it is C. Ulla says it is C. What do you think is the answer for this? What do you think is the answer for this? Okay, Mahantesh also says it's C. Any newcomer out there probably want to try out giving the answer for this? So C is what? C. Statement 1 is incorrect, but statement 2 is correct. Let me check it out. In Viola, Comolina, Oxalis, Viola, both autogamy and geitonogamy is prevented. Arey Baba, they have clistogamous and they have chasmogamous flower. Autogamy can happen, geitonogamy can happen, any gamy can happen. So this is obviously going to be wrong because it is saying that it is prevented. Wrong statement. Geitonogamy is similar to autogamy. Is it similar to autogamy? Not exactly, but only in one case. Since the pollen grains come from the same plant. Yeah, in that case, yes. So genetically, I'll say it's similar to autogamy. Why? Because the same plant parts are coming together. Genetically it's same, technically it's not the same. But anyway, it is same because they have mentioned since the pollen grain comes from the same plant. They had explained this. So this statement is correct, the other statement is wrong, and you had got the correct answer. Viola is clistogamous flowers. Manoj, Viola has both clistogamous as well as chasmogamous flowers. Both of them. Okay? So Comolina, Oxalis, Viola, they have both open as well as closed flowers, at least according to your NCERT. Fine. By the way, one more thing. For this, I spoke about castor, right? I spoke about castor. Castor is like this. Similarly, maize is also like this. Chief Minister, CM, you can remember. Monoecious plants with unisexual flowers. This is a bisexual plant, but with unisexual flowers. This is a unisexual plant with unisexual flower, dioecious. Okay, so anyways, let's go ahead with the next question then. Guys, if it's okay, if it's cool, can you give me a thumbs up? If you're understanding it and I can go ahead peacefully with the next question, can you give me a quick thumbs up? Okay, perfect. Thank you. Which is not a characteristic feature of wind-pollinated flowers? Which one is not the characteristic feature of wind-pollinated flowers? Feathery stigma, presence of single ovule in the ovary, pollen grains are non-sticky, and flowers are very colorful. It's a very easy question. Come on. Which is not is being asked, okay? Which is not a characteristic feature of wind-pollinated flowers? Manoj says it is D. Ullas is saying it is D. Okay, both of you guys are saying it's D. And I think Mahantesh is also saying it's D. So I have to log this up. The answer for this question is going to be D. Listen to me. Use your brain. It's very, very simple and you have already done that. Okay, so this could be, I'm telling you, this MCQ question could be coming in a 2-marker question. And they can ask you, what are the features of wind-pollinated flowers? As simple as that. So please know all the things that are given over here are important. Wind pollinated over here, in the case of wind pollinated, the pollen grains which are there, do you think if at at all they are sticky, they are going to land up somewhere over here. They have to be going via wind, but they will be landing up over here. I'll say that I am not able to move. Always remember, pollen grains have to be non-sticky. So this is the correct one. Now feathery stigma, these non-sticky pollen grains, they are just moving around. You need someone to trap them up. They cannot come down. They need someone to trap them up, which is why you need feathery stigma. So this is also correct. And I'll tell you one more thing. The pollen grains are released from the anthers. You would be having exposed anthers. What do you mean by exposed anthers? The one which are long and everything, so that when the wind comes, they can carry the pollen grains along with it. That is why they will be having exposed stamen. Exposed stamen, always remember that. Finally, this is something that is difficult to remember, but still try. Presence of single ovule in the ovary. Why? Listen to me. If ovary has multiple ovules, and this is wind pollination, this is not even insect pollination, there are chances of failures. My chumma I am wasting my ovules for this particular purpose. No, I will be chumma wasting for my ovule for this particular purpose, which is why I'll make sure that there is only one ovule. There is going to be only one ovule per ovary, such that my energy is not being lost. My energy is not being lost. Got it? So presence of single ovule in ovary because of the same reason, and the flowers are very colorful. That is usually the case of insect pollinated flowers. Insect pollinated flowers, not wind pollinated. Got it? So I hope you remember the features and you will write it up. Okay guys, I have a question for all of you. Okay, ready? 1, 2, 3. Do I have it over here? I do not have it over here. So I'll ask it to you directly. Tell me the name of the plants, 2 plants, that is going to undergo wind pollination. The examples of 2 plants that undergoes wind pollination. Any guesses are accepted? Come on. Maize. Okay. Corn cob and grasses. Exactly. So in your NCERT, they have given it as corn cob and grasses, and please do remember it. Please do remember it. Very, 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 very important. If at all you get it wrong, probably that's the only example that you have to remember for wind pollination. So do remember it. Okay? Good one, you guys. Good one. Now, let's go ahead with the next question, and it goes like this. Wind pollination is common in. Wind pollination is going to be common in. Now, you've already got the answer. Still, you can try. Still, you can try. Just a second. C, right? So C is going to be the answer, grasses. So last said maize first, that is the same thing. Corn cob is the same thing. So you guys are correct. Okay, creative. Hi ma'am, from Mangalore, your classes are literally so good, which clears all our doubts. Thank you so much, creative. That's so sweet of you. Thank you so much. I have taken a screenshot of it. It feels really good. Anyways, guys, the answer for this question is going to be grasses. Do not forget it. Okay. If all this thing is clear to you, can I move to the next question? Can you give me a thumbs up? If all of it is clear to you, can you quickly give me a thumbs up? I'll move on to the next question. Yes, perfect. The next question that is there, double fertilization is. Now you will be able to answer this question since we have spoken about it before. Fusion of 2 male gametes of a pollen tube with 2 different eggs. Fusion of 1 male gamete with 2 polar nuclei. Fusion of 2 male gametes with 1 egg, syngamy and triple fusion. What do you think is the answer for this particular question? Ullas says the answer for this question is A. Sure? Are you sure? Wanna recheck it once more? Double fertilization is what? Okay, Ullas now says it's D. Yes, that's the good thing. That's the good thing. Okay, creative says it's D as the answer. Perfect. Perfect. Anyone else want to give a wild guess out there? The answer for this question is going to be, Manoj also says D, which is why I'll lock it as D. Double fertilization, we know that it is going to be the first fertilization or the fusion is what? With the egg cell and the female gamete. Egg cell and the male gamete, that is known as syngamy. Done. The next one is basically between the male gamete and the two polar nuclei, which is known as the triple fusion out there. That is also done. Why these options are wrong? Fusion of two male gametes of a pollen tube with two different eggs. Arey Baba, we just have one egg. How can we say two different eggs? Fusion of one male gamete with two polar nuclei. That is the just one fertilization. No, that is triple fusion. You need syngamy also. Fusion of two male gametes with one egg. Why will you do that? That will also be wrong. So which is why the answer for this question is going to be D. The answer for this question is going to be D. Ulla, swiping mistake, lol, my bad. It's okay, bacha. Okay, so that's all. Let's go ahead. Okay, so two male gametes, one coming for this, another coming for the polar nuclei out there. Guys, if it's clear, can we move ahead with the next question? A quick thumbs up. A quick thumbs up. My cheeks are paining after smiling so much. Let's go. Okay, perfect. In a fertilized embryo sac, the haploid, diploid and the triploid structures are represented by. Now this is a very important question. In a fertilized embryo sac, the haploid, diploid and the triploid structures are represented by what? You can take your time for this question, but this is very, very important. Manoj says the answer for this question is going to be A. Okay. Anyone else with a other guess? Anyone else with any other guesses out there? Okay, Akash is there. Hello, Akash. Akash also says the answer for this question is A. Okay, now since there are two people out there, I'll say yes, it's A. But let's check it out, man. Ullas is also saying it's A. So we have to double lock it anyways. So listen to me carefully. The embryo sac containing haploid, diploid and triploid structure. That is the question to you. So over here, if at all we see, this is the embryo sac that we have. At the top, you have the antipodal cells. You know that all of them are going to be haploid. Please remember, they are going to be haploid. Okay, what are those? They are antipodals. The one that does not change the ploidy, I'm going to put it in yellow, such that you have a photographic memory of it. Okay, three of them are going to be haploid. They are synergids. They are also going to remain as haploid cells. Synergids, haploid. Okay, the one that changes its ploidy is right over here. First one is going to be your egg cell itself. First one is going to be egg cell. It was haploid first, but then when it fuses with the male gamete, it is going to form something that is known as zygote. And since the male gamete contains haploid nucleus, it also contains haploid, it becomes diploid. The ploidy changes. The ploidy changes over here. Okay, now we have one more thing over here that changes its ploidy, and that is going to be what? Your central cell. So this is your central cell right over here. It was having two nuclei. So central cell initially was diploid. Why diploid? Because two nuclei is there, n plus n, it becomes 2n. Now the male gamete also comes and fuses with it. Male gamete comes and fuses with it. It is haploid in nature. So haploid plus diploid, it becomes triploid. What triploid? What? Primary endosperm nucleus. The nucleus that all the three nucleus are going to come together and form is known as primary endosperm nucleus. Always remember that, and it is going to be triploid. So the question to you guys is what? Which of them is haploid, diploid and triploid? So haploid, okay, after in a fertilized embryo sac, that means fertilization has taken place. So haploid has to be either synergids or antipodals. In all this option, the first one is synergids or antipodals, so I cannot rule it out. Okay, but the triploid structure will always be one thing only, and that is going to be the primary endosperm nucleus, or basically the primary endosperm cell. So the last option that you have to look is this and this. Okay, you can cancel these two options. And now you have got these things. The diploid one obviously is going to be zygote. It cannot be synergid. So which is why the answer for this question is going to be A. Here is a list of all the ones, okay, which are going to be haploid, diploid and triploid and everything. I have a question to you. Okay, can I ask this question? Akash, hello. And Creative, yes, Creative also gave me the correct answer. Now the question is, the cells of the nucellus. The cells of the nucellus, if at all you remember nucellus. Within the ovule, you had the cells of the nucleus. What is their ploidy? Guys, can you tell me what is their ploidy? The cells of the nucleus, are they haploid? Are they diploid? Are they triploid? What's the scene with them? Manoj says it is going to be diploid, 2n. Okay. What about others? What about others? Okay, Ullas says it is going to be diploid. Exactly. Listen to me very carefully. This is just to give you, what do you say, cause confusion in your head. Every other cell in a plant, apart from the gametes and also the synergids and the antipodals, every other cell is going to be diploid in nature. Normal cell is going to be diploid. Okay, so yes, Ullas, yes, Manoj, yes, Creative, all of you are correct with that. Perfect. I just wanted to check that out. Okay, now we are coming. Ayyo, what is this? I showed you the answer, but still you can try. Endosperm persists in the mature seed in what? Groundnut, castor, pea, or beans? Endosperm persists in the mature seed in what? You have got these many options. Okay, B. Manoj says B. Ullas also says B. What is B? Castor, Baba. Okay, now listen to me very carefully. I can twist the question and I can ask you. I'll ask you a question. Since this question has been a flop, I'll ask you a question, okay? Because you saw the options, right? The option goes like this. So you have, I'll ask you, endosperm does not persist in what? Okay, that is a question, and I'm going to change the options. It does not persist in okay. Can you tell me what is the answer for this? Sorry, ma'am, I cheated. Oh, it's okay. Everyone did, don't worry. Endosperm does not persist in, and you have all the options: groundnut, castor, wheat, and maize. What do you think will be the answer in that case? Manoj says the answer for that question would be A in that case. Okay. What about the others? Is it going to be A? Endosperm persists in the mature seed. Ullas also says it's A. Exactly. So listen to me carefully. You have endospermic and non-endospermic seeds. Okay, and it's pretty simple. Very, very simple. So primary endosperm cell that you made after double fertilization, no? That cell is going to divide, and you get the whole endosperm tissue. That is what you get. Endosperm tissue is being formed at the end. Now this endosperm tissue which is being formed, Baba, okay, in some cases it is just staying there. It just stays there. Why do you think endosperm is being formed? Because over here, the zygote which is being formed, it has to transform into the embryo. It has to transform into the embryo. Now for that, the zygote needs nourishment. Who provides it? The endosperm. So there are some embryo which says that, okay, I do not want your help anymore. I can take care of myself. I do not need any endosperm now. I'm going to take all the endosperm with me and I'm going to make 2 cotyledons for myself. So there are going to be some embryo which is going to have 2 big cotyledons right there. This type of embryo, basically I'll say, is found in dicots. Found in dicots, they take the endosperm and convert it to 2 cotyledons out there. Usually they are found in the case of dicots. In this case, do you think endosperm will be left behind? No endosperm is left behind. Everything is utilized. I'll say that they are non-endospermic, or basically I can also call this as non-albuminous. They are non-albuminous. Why? Because no endosperm is left behind. Why no endosperm is left behind? Because they have utilized it for the formation of cotyledons. Then there is another case where the embryo says that I really appreciate everyone helping me right now. I want the endosperm to be still there. I'll make something for myself. Okay, I'll be having 1 cotyledon for me, but I'll also have little bit of endosperm left behind in me. Here in this case, only 1 cotyledon is being made. I call these plants as monocots. Okay, and I'll say that these seeds are albuminous. Why albuminous? Because over here some amount of endosperm is left behind. Okay, so usually you can remember it like this. Please listen to me carefully, very important thing. Usually you can remember every monocot which is there, it is going to be albuminous, and almost all the dicots are going to be what? Non-albuminous, except one guy. So there is one dicot. Okay, there is one dicot. Where is that? And can you name the dicot? Can you name that dicot which is a dicot but still comes under albuminous category? It's not a monocot. It comes under albuminous because it has little amount of endosperm left behind. Can you name that particular dicot that has endosperm left behind? Any idea? If you guys are able to name them, then I'll give you a Kit Kat. And you'll be thinking, ma'am, Kit Kat, who wants Kit Kat? But still, you can give it a try. I'm asking you a question. I'm putting this question again in front of you. There is a dicot. Usually all the dicots which are there, they are non-albuminous. That means they take up all the endosperm and make cotyledons out of it. But there is a dicot. There is a dicot which is going to be albuminous. Which is going to be albuminous. P, no, not P. By the way, dicots are the one which you can break into two. So pea, groundnut, everything, they come under dicots. You can just imagine like that. But anyway, P is not the answer. But good guess. I'll tell you. Ullas, yes, said. And Pratiksha is there. Pratiksha came up with a bang and said that castor. Okay, so Ullas and Pratiksha, you guys are correct. So actually, it's the castor that we are talking about right over here. It's a dicot, but it comes under albuminous category. It's a dicot, exception. I'm writing exception. Dicot that comes under this is known as castor. Apart from it, it's just monocots. What are monocots that you cannot break into two? For example, wheat. For example, what other examples that you have is wheat, maize, rice, everything that you cannot break into two, they come under monocots and they are albuminous. Here, what you can break into two, they are dicots and mostly they come under non-albuminous. You have pea, you have groundnut, you have beans, all of them. Got it? Clear? Endosperm persists in the mature seed of castor, and rest three of them, they are dicots. They are all dicots, but this guy, even being a dicot, it is albuminous in nature. If it's clear, can you give me a thumbs up? If it's clear, super clear to all my cutest little children, can you give me a thumbs up? Thank you so much. Okay. Now, let's go ahead with the next question out there, and that is this. Come on, try it out. Coconut water from a tender coconut is what? Coconut water from the tender coconut is what? Ma'am, for biology mock test, I watch your sessions daily, which help me score good marks in biology mock. I watch your classes every day, literally made me easy to learn biology. Thank you so much, creative. Thank you so much. Okay, so Akash says the answer for this question is D. Deepak Reddy is there saying it's 4. And the cute YY is saying it's 4. So everyone, Ullas is also saying it's 4. Let's go. 4 is what? Free nuclear endosperm. Is that so? Yes, Baba. All you guys, when you drink coconut, always remember the white one which is there is nothing but the nucleus which is covered by the cell wall, which is why they have been cellularized. They are all together, they have been cellularized. Now there are some nucleus which says that no one can catch me, I do not have the cell wall. These free nuclear cells which are there are known as free nuclear endospermic cells. Okay, the one which has cell wall, it turns into this. So that's the logic. If you generally see a coconut which is there, when you go and say, uncle, please give me a coconut water and things like that, they'll check. Some of them will be just having the white kernel, some of them will not have water. What do you mean by that? That means that these free nuclear endosperm which were there, these cells are being caught and their cell wall has been made around it. That is why they cellularized over here. Okay, and usually, ma'am, talk slow. Okay, I'll talk slow. So usually it happens like that. So after some time, as and when it is growing, my dear children, all this free nuclear endosperm is going to be taken up to the formation of the cellular form of endosperm that you eat. Kernel is what you call it. Okay, the cute. Okay, you speak very fast, Akash. I hope now it's clear. I'll slow down. Okay. So everyone who gave me the correct answer, guys, you are doing awesome. So coconut shows both stages, free nuclear water and the cellular form which is known as kernel. If that is clear, can you give me a thumbs up? I'll go on to the next question. Perfect. Thank you. The next question goes like this. In In a monocot embryo, shoot apex and the leaf primordia are enclosed in a hollow foliar structure called what? Pericarp, coleoptile, endosperm, or basically coleorhiza. What do you think is the answer for this? In a monocot embryo, shoot apex and the leaf primordia are enclosed in a hollow foliar structure known as what? Ma'am, for NEET, according to you, which competitive books you prefer for biology, ma'am? I'll say MTG and also NCRT. MTG for question practice and NCRT for every theory part. Okay. Guys, answer for this? Any idea? In monocot embryo, shoot apex and leaf primordia are enclosed in a hollow foliar structure that is known as what? Don't know, ma'am. Manoj says don't know. Give the first time. Ullas says B. Akash also says it is B. Let's check it out, okay, together. For those of you who do not know, well, the answer for this particular thing is going to be coleoptile. It's going to be coleoptile. But I really want to help you over here. The reason being that this structure, monocot embryo structure, is a 3-marker question. And everyone goes nuts. And everyone basically goes nuts. If at all you are ready, I am going to explain the 3-marker question right over here because that is definitely going to help you. Anyway, I was planning to talk about that here. 3-marker question. Draw a neatly labeled diagram of LS of embryo of grass. Grass is nothing but the monocot. Grass is nothing but the monocot. So can I explain this diagram right now? Pratiksha also said the B is the correct answer. Perfect. Can I explain this diagram right now? Is it okay for you? If you get to know the diagram, you will be able to solve that question and will not have to repeat the diagram again later on. Yes, cool. Listen to me carefully. Very simple logic is there. Okay. So there are two type of seeds. There are two type of embryo, I can say. Dicot and monocot. What happens in dicot? Baba, in dicot, you will be able to see something like this. Here, you have the axis. This is known as embryonal axis. That means this is the one that is going to form a stem later on and also the root later on. This whole part. What do you call this as? Embryonal axis. Embryonal axis. Every other embryo will be having it. Dicot, monocot, everything. Now in this embryonal axis, there are 2 cotyledons which are there. 2 cotyledons which are there. That is why I called it as dicot. What are these cotyledons? Cotyledons are basically made from the endosperm that was there. These cotyledons are going to help in the embryonal axis to grow. This portion over here, the down portion over here is known as radicle. It is going to grow into what? The root. It will be growing into a root later on. Okay, and since it is going to form a root has to go within the soil out there, which is why it will need some protection. It is going to need some protection. I'll say the protection is given by the help of the root cap. Cool. Now, who is providing energy? The cotyledon is going to do all the work. Okay. Now, ma'am, this is about the root. What about the shoot? So, this top portion that you see over here, this is known as plumule. Plumule is the one that is going to give rise to the whole shoot setup. The whole shoot setup. This whole thing that you see over here is a dicot embryo. It's a dicot embryo which is there inside the seed, which is going to be there inside the seed. Now, listen to me very carefully. The point of attachment that you have here of the cotyledons, below it, this particular whole region is known as hypocotyl. Hypocotyl. Hypo means less. So, below you can remember hypocotyl. The point of attachment after this is known as epicotyl. This is known as epicotyl, and this whole thing is the structure of the dicot embryo. If at all you have been asked about the structure of dicot embryo, you can either draw like this or you can draw in the manner that is given in your NCRT. What have they done? They have made something like this. Okay, so this is going to be, here is your one cotyledon. The next cotyledon is going to be like this. They are just sleeping out there. So these two are the cotyledons. This is your hypocotyl. This is your epicotyl. Over here you have the radicle, and then you have to make the root cap. You have to form the root cap. Here you have the plumule, and that's all. This diagram and this diagram is technically the same. This is what your dicot embryo. Is the dicot embryo clear to you? This can also come as a 3-marker question. That is why I'm telling you. If it's clear, can you give me a quick thumbs up? I'll talk about the monocot. The reason I was talking about this first is because you need some understanding. Otherwise, you will not technically relate both of them. If it's clear, guys, thumbs up. Okay, Manoj has given me thumbs up. Only Manoj understood. What about others? Children, understanding? Not understanding. Okay. Cool. So now I'm going, okay, the cute SSSY. Hello ma'am, I completed my 12th from your videos only. I just got notification now, so I joined here to teach. Awesome, Jasmine ma'am. Thank you so much for your teaching and your motivation. I got, oh my God, that's so sweet of you. And I'm so happy for you. Congratulations, first of all. And so grateful for you to come back and talk about it over here. I hope that would increase a little bit of trust in the children who probably have joined this year. So thank you and God bless you, and I love you a lot. Please take care of yourselves. Okay, now we are going to come over here. Ma'am, what is polyembryony? Akash, I'll come to that question. Okay, don't worry. All those questions will be covered over here in this video itself. Okay. Love you too, dear. Okay, let's come back now. So this is about the dicot embryo. Now please take a screenshot of it because it will go. I do not have the page for this, okay? Over here, now I'm going to make your monocot embryo. This thing is very simple. In monocot embryo, what is going to happen? Here you would be having the embryonal axis. You would be having the embryonal axis, but this time you just have one cotyledon. You do not have two cotyledons, rather you just have one cotyledon, and that cotyledon is right over here. And whenever you have only one thing, do not have multiple things, you just have one thing. So I'll say that basically I'll name it. I'll call this as scutellum. Ayo, I should put it somewhere else. So sorry children. So sorry because I need it for labeling. Okay, I'll call this as, come on, work. Scutellum. This is the only cotyledon that the monocot has. This is the embryonal axis. This part is going to be the plumule. This is going to be the plumule. Here I have the radicle. Here I have the root cap. Same like that. The only portion is that, only difference is that it does not have the cotyledon. The next difference that it has is what? The next difference it has is basically there will be a little amount of endosperm left behind. There will be little amount of endosperm which is going to be left behind. That is also right over here. Now, the differences that you have between monocot and dicot is not just that. The next difference that you have is covering the plumule, or basically the shoot apex. Okay, the shoot apex that is going to give rise to the shoot. Baba, over here you would be having what? You would be having one very important thing which you call as coleoptile. Okay, it is known as what? Coleoptile. Always remember P over here and P over here. Covering the plumule or the shoot apex, you have the coleoptile. Similarly, over here, covering the radicle or the root cap, you have coleorhiza. You have what? The coleorhiza. R over here, R over here. You can remember like that. It's very, very important. Now, this is not the diagram that is given in your NCERT that makes you go nuts. Ma'am, what will I do? What will I do? So listen to me carefully. I'm going to make a diagram over here, which is exactly going to be similar like this, almost. And you can draw it in your examination as well. You have to make a whale head. Okay. There is a fin of the whale. There is a tail for the whale. And then there is a smaller fin right over here. Okay, so whale, and then you have two fins and then two tails. Here you make a triangle like this and color it up. This is going to be your coleoptile. Okay, make a smaller one inside it. This is going to be your shoot apex, or basically plumule. I'll write shoot apex because that is how it is given in the NCERT. Shoot apex. Okay. Now this, both of them join together, make a line and let it go over here and let it come back. Let it go to one tail and join it back over here. I've made it go, come back and go, went over here, and then it has joined back over here. First of all, if this is the embryonal axis, this part is going to be what? The scutellum. This is your cotyledon. This is your cotyledon. Okay. Now over here, what will you have? You will be having your radicle. Covering the radicle, what will you have? You will have the root cap. Again, make a triangle, cover it up. What do you have? This is known as the coleorhiza. This is known as the coleorhiza, and that's all. That's the only diagram that you have to make. Okay. Ma'am, this diagram, how many marks? How many marks? 3 marks. Okay, and it's a very, very frequently asked question. That is why I'm telling you, please try to draw it. Now, in the question that you were being asked, the question was, what was that something that covers the shoot apex? It is going to be known as coleoptile. And for children who forget, that's what I'm telling you, PPRR. Remember like that. Got it? Got it? Is it clear? Everyone? So we have basically technically solved a 3-marker and a 1-marker question altogether. If yes, can you give me a thumbs up? Creative ma'am, you're so friendly and soft spoken, but our biology ma'am shouts at it. Ayyo, I'm so sorry that happens with you, but I'm really glad that we met. Okay, so okay, Manoj says it is clear, and I hope it's actually clear to all of you. In that way, you have got your 3 marks, and I'll come back to this. Oh my God, my back is paining. We have almost done, guys. Where are we? Yes, we were here. Monocot embryo, shoot apex and the leaf primordia are enclosed in a hollow foliage structure that is known as coleoptile. Done. Now we go ahead with the next question that is there, and it's like this. Perisperm differs from endosperm in what? Perisperm differs from endosperm in what? Being a diploid tissue, being a haploid tissue, having no reserve food, is a formation by fusion of secondary nucleus with several sperm. What do you think is the answer for this? Perisperm differs from endosperm in what case? If you do not know what is perisperm, let me tell you. Sometimes inside the ovule, inside the ovule you have the nucellus, right? And nucellus is generally being utilized. When the embryo sac is being formed within it, the nucellus is generally being utilized for providing nourishment for that. But sometimes the nucellus persists as such. Why? Because later on it waits for the embryo to be made. It nourishes the embryo as well. Not just the embryo sac, but the embryo also. That persistent nucellus is known as perisperm. So I can say the cells of the nucellus forms the perisperm. Now you can give me the answer. Creator says the answer for this question is A. Come on, that's your senior. Now juniors, come on, give me the answer. Perisperm differs from endosperm in what? You know, endosperm is what? It is going to be triploid in nature. Is it going to be triploid in nature? Yes. What about perisperm? It is made up of what? Cells of the nucellus. The cells of the nucellus, and the cells of the nucellus are what? It is going to be, Akash is saying A is the answer. Yes, the cells of the nucellus are going to be diploid. No, normal cells they are going to be diploid. So perisperm differ from endosperm in nature in being a diploid tissue. Not being a haploid tissue. No, they are not haploid. Having no reserve food, Baba, they have reserve food. Okay, it's information by fusion. No, that's not happening. So only answer for this question is going to be A. Ullas, ma'am, please update about NEET counseling through KI gave KCET. There are no updates. So there are no updates that have come yet, dear. I do not know while I am in the live if there are updates. If yes, I'll definitely let you know. Okay? Creative, you're also junior. Oh, sorry. Okay, so yes. So the answer for this question is going to be A. Children, if you have got it, can you quickly give me a thumbs up? If you have got it, quickly give me a thumbs up. We'll move on to the next question. Okay, thank you. Let's go ahead. Non-albuminous seed is produced in, come on, we just discussed this. Non-albuminous seed is produced in maize, castor, wheat or pea. Non-albuminous seed is produced in what? Maize, castor, wheat or pea. Akash, thank you. Thumbs up. Okay. Ullas definitely would be doing that, dear. Definitely would be doing that. Okay, Manoj says D is the answer for this question. Non-albuminous seed, D. Yes, why? Because non-albuminous are usually what? Dicots. What do you mean by non-albuminous? They do not have endosperm left behind. Why? Because they take it up for the formation of cotyledons. And 2 cotyledons means nothing just going to be left behind. So Akash also gave the answer as D. Creative also said it is D. The answer is D. These 3 things, they are going to be albuminous. This is a dicot, but this is an exception. Okay, so we have discussed about it and it's done. Now, which part of the fruit labeled in the given figure makes it a false fruit? Which part of the fruit labeled in the given figure makes it a false fruit? So you have you have C over here, you have A over here, you have D over here, and you have B over here. So this is technically, B is technically the seed. Rest all of it is given over here, and you have to tell me. By the way, I hope you're able to see it. This is going to be C, this is going to be A, this is going to be B, and this is going to be D. Okay? Manoj says the answer for this question is going to be D. What about others? Akash also says it is D. Okay, everyone says D, 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 D. So then we have to solve D. D is the answer. D is going to be thalamus. Yes, it is going to be thalamus. The reason being that is that, listen, fruits, true fruits are the ones where basically, the ovary which is there, it transforms into a fruit and the ovule transforms into the seed. Basically, the ovary should be transforming into the fruit, only the ovary. But in the case of false fruit, it's not just the ovary, it could be some other floral parts as well. For example, suppose you have, when you talk about a flower, okay, when you talk about the flower, it is going to be there having a thalamus, then a pedicel out there. Suppose this thalamus is also contributing in the formation of a fruit. Apart from the ovary, this is also contributing in the formation of a fruit. Do you call this as a true fruit? No. You will call this as a false fruit. Okay, examples for that, guys, can you guess? Examples for false fruits? So over here, they had given that A is basically ovary. A is basically mesocarp. That is a part of the ovary. B is endocarp, again the part of the ovary. D is going to be the seed. That is basically the ovule. It has nothing to do with true or false fruit. Last is C. C is the thalamus they had mentioned. Thalamus here is participating. That is why it is known as false fruit. Now I'm asking you a question. Can you give me an example of the plants which are generating false fruits, which will be having false fruits? Do not know? You have apple. Okay, you have apple, you have cashew, and you have strawberry. Three things. Okay, apple, cashew, and strawberry. Apple and strawberry. Perfect, Manoj. Okay, cashew is also there. These three plants are there given in NCERT, which is an example of what? False fruit, strawberry. Perfect. In all this case, different other parts are also participating. We will be getting to know more about it while we go ahead. Now, I think this is going to be the second last question. Or the third last. Okay, let's go. MCQ question. Okay, read the two statements and choose the correct option. Apomixis is the production of seeds from the unfertilized ovules. Embryos produced from apomictic seeds are generally identical to the parent plant. Generally identical to the parent plant. Akash says the answer for this question is going to be A. How so fast, huh? Apomixis is the production of seeds from the unfertilized ovule. Is this the correct statement? Embryos produced from apomictic seeds are not generally identical to the parent plant. So only one answer, A. Only one person answering this question. Guys, I'm going to move forward and probably talk about it because I think it's an important question and this has come before. So over here, the question goes like this. Just a second. I'm just trying to find the question itself. Apomixis, yes. This is a 3-marker question again. I hope you mind explaining it. Apomixis and polyembryony, everything I'll explain it right now itself. Works? Okay, listen to me very carefully. Then probably we can come back to that MCQ. What do you mean by apomixis? Apomixis is the case where suppose over here you have the ovary. Okay, this is going to be the ovary. Inside the ovary, what do you have, Baba? You have the ovule, right? Ovule. Now, inside the ovule, what do you usually expect? The embryo sac will be there. And in the embryo sac, the egg cell has to be haploid and the synergids are haploid, the antibody cells are haploid. This is how it usually goes, generally. Now, when the egg cell, general case I'm talking about, okay, generally what is happening, the egg cell which is going to be haploid fuses with the male gamete, which is again going to be haploid. Together they come together to form a diploid thing that is known as a zygote. And the moment it forms a zygote, I will not call this as an ovule anymore, I will call this as a seed. I'll call this as a seed because zygote is being formed out there. Now here comes the difference. Okay, here comes the difference in apomixis. What is going to happen in apomixis is that within this particular structure, Okay, there are two ways apomixis can happen. There are two ways apomixis can happen. I'm talking about the first way that is happening. Here, the female gamete which is there, that is the egg cell which is there, it is going to be diploid in nature. It is not haploid. Rather, the egg cell which is there, it is going to be already diploid in nature. Ma'am, how can that be possible? It can be possible if at all it didn't undergo meiosis. If there was no meiosis, basically the number of chromosomes remains the same, just like a normal cell, it is going to remain diploid. Now here it is already diploid. Do you think you will need a requirement of a male gamete which is haploid to come and fertilize? No. There is no requirement of the male gamete to come and fertilize. That means I can say that there is no requirement of fertilization at all. No fertilization is required over here, and without fertilization, this egg cell which is already diploid in nature transforms into the zygote, and the zygote later divides to form the embryo. So Baba, since you had anyway formed the zygote over here, anyway formed the embryo over here, can I call this ovule now as a seed? Yes. But did you see any sort of fertilization happening up over here? No. No fertilization, ma'am. Simply one thing happened over here. No meiosis took place. Egg cell was already diploid in nature, which is why it did not need fertilization to form the zygote or the embryo. In this case, when you are forming a seed without fertilization, you will say that this is known as apomixis. This is known as apomixis. This is one way of apomixis, okay, where the meiosis is not happening. There is one more way for this to happen. The other way is that again you have the ovary, again you have the ovule, here you have the embryo sac. And if you remember, I told you sometimes this nucellus, the cells of the nucellus, they persist. The cells of the nucellus, they persist. Now these cells which are persisting over here sometimes can get inside what? The embryo sac. And when they get inside the embryo sac, always remember these cells which are there, the nucellar cells which are there, they are already diploid. They are normal cells, so obviously they have to be diploid. They are getting inside the embryo sac. Once they are inside the embryo sac, what are they going to do? They are going to form the embryo. They will be forming the embryo. And do you think they are going to form just one embryo? No. If you think like that, you are wrong. They are going to form multiple embryos. But anyway, we have formed the embryo. Same thing. We have formed the embryo over here also. Will I now call this as a seed? Yes, I will call this as a seed. Do you require fertilization there? You do not require fertilization there. So either it can happen in this way or it can happen in this way, but apomixis can happen. If it is happening in this way, you need to note one more very important thing. Baba, you have formed what? Multiple embryos out there. You have formed multiple embryos out there. This process of formation of multiple embryos is known as what? Polyembryony. This is known as polyembryony. Got it? Very, very simple. Now, it will not always happen when apomixis is happening. Sometimes it happens. Sometimes it happens, and it's usually seen in citrus fruits. It is usually seen in citrus fruits. For example, you're talking about mango. If you want to talk about oranges, in those cases, you will be seeing polyembryony, where multiple embryos are being formed. Okay, do not think that these two they're always together, happening together. No. If here apomixis is happening like this, there is no multiple embryos that are being formed. Here, when the cells of the nucleus are involving, yes, polyembryony has been taking place over here. Example for that is basically mango and oranges, where inside the seed you will be finding multiple embryos. Cool. And generally, the example of apomixis is basically just two in number. One is grasses. You're going to see them in grasses, and apart from that, you're going to see them in plants belonging to Asteraceae family. Ga, okay, Ga. This is something very, very important. Got it? I hope it's done and dusted. So yes, and now one more very important thing here, since fertilization is not going to happen, there is no fertilization that is taking place over here, which is why I want to remind you one thing. If no fertilization, then they are going to be exactly similar. For example, the plant that is going to be formed is going to be exactly similar like the female parent. So I'll say it is going to produce clones, and this is considered to be asexual reproduction, even though it is mimicking sexual reproduction. Why it is mimicking? Because over here zygote is being formed. Zygote is an end product of sexual reproduction, no, but here it is just mimicking sexual reproduction. Actually asexual reproduction is happening over here. I hope this is also clear to you. Ma'am, important questions for 3rd and 4th because I have bio test tomorrow. Manoj, you're talking about 3rd and 4th chapter here? Are you talking about 3rd and 4th chapter? If yes, I'll request you to just browse through YouTube. You would have to look for the previous videos that we have done. Okay. If you go around, you have to go back a bit and you have to see PU2 final fixed question video. There you will be getting the fixed questions of these chapters as well. Manoj, can you do that? Okay, or if you wait until the once the live is ended, I can probably, if you comment down, I can provide you the link for that. Okay, so this is about it. Now there is one more part of this question that is mention its advantage to a farmer who uses them. Mention its advantage to the farmer who uses them. First of all, listen to me. Farmers which are there, okay, farmers who are there, they do what? They take hybrid seeds. Hybrid seeds means, for example, I have a plant like this, I have a plant like this. This plant is good in terms of pest resistant. This plant is good in terms of drought resistant. I make hybrid seeds out of it. But the problem of these hybrid plants that are going to grow up is that they are again going to undergo sexual reproduction. And when you undergo sexual reproduction, Baba, not all features are going to be the same in the new plant that is going to be there. Sexual reproduction brings variation. Okay, so when you talk about hybrid seeds, first of all, it needs a lot of money to buy them. The second thing is that if at all you let them grow, they will undergo sexual reproduction, and basically the next generation will not be similar to this. So you are going to see segregation of the character. New character is going to be formed. It can be in favor for you, it may not be. Right? So instead, what if I use apomictic seeds? Apomictic seeds, the good part of the story is that basically over here you just require one plant. This one seed goes, forms the entire plant out there. The progeny will also be similar, just exactly similar to this particular plant. So if I'm assuming this plant has all the good features, it is going to remain the same throughout. Why, Baba? Because it is not undergoing sexual reproduction, rather it is undergoing asexual reproduction. So the good part of the story is that basically it becomes cost effective. You do not have to buy hybrid seeds, you just have to buy the apomictic seeds. That 2 for 1s. Next thing, the hybrid vigor is maintained. That means all the important features which are there, the major features that you were looking forward for, it remains the same. Why? Because there is no sexual reproduction, only asexual reproduction is taking place over here. Okay, next thing, if I want all the plants to be uniform, obviously they have to be coming from the same parent. That is also being guaranteed. So there will be uniformity, it will be cost effective, and hybrid vigor is also being maintained over here. So what are some seeds? Why are some seeds referred to as apomictic seeds? Some seeds are called apomictic seeds because they are formed without fertilization, that is fusion of gametes. The embryo develops without meiosis and syngamy, producing seeds genetically identical to the parent plant. You can write this and you will get your marks. Is this question clear to you? Every one of you who is watching this video, Baba, the question is clear? Understood? Perfect. Cool. Now over here I have a question as well. So we were solving actually this question out there. Right? Apomixis is the production of seeds from unfertilized ovule. Is that particular statement true? Unfertilized ovules, that is correct. No fertilization is there, so this is correct. Embryos produced from apomictic seeds are not generally identical to the parent plant. That is wrong because they are identical to the parent plant. Here not is written. So I'll say statement 1 is true and statement 2 is going to be false. That means the answer for this question is going to be A, as someone said before. Cool? Perfect, Manoj. Now, I think this is the last question that we have. The picture given alongside is an example of what? This is the picture that is given to you and you have been asked what is happening over here. Is it multicarpellary apocarpus gynoecium, multicarpellary syncarpus gynoecium, monocarpellary gynoecium or apocarpus gynoecium? I am right. Yes, Akash, you're there. Yes, definitely you are right. Okay. Manoj says the answer for this question is going to be A, multicarpillary apocarpus gynoecium. Is that so? My name is Jasmine, dear. Okay, so yes, guys. Manoj gave me the answer as A. Okay, Akash is giving me the answer as B. B is what? Multicarpillary syncarpus gynoecium. So listen to me carefully. Actually, this particular plant looks like this. It looks like this. Over here, this is the stigma, and you would be having the style, and all the stigma, there are so many stigma and styles out there. That means they are definitely multicarpillary in condition. Okay, but the ovaries of all of them, they are going to be fused up together. They are going to be fused up together. This is known as syncarpus condition. This is not apocarpus, this is syncarpus. Okay, and this is seen in the case of papaware. Papaware is a plant from where you get opium and all things like that. Opium is a drug. Okay, so this is papaware from you get it, and Baba, I am gonna make it over here. So this is basically, I can say it is multicarpillary, and it is going to be syncarpus gynoecium. Syncarpus, not apocarpus. Apocarpus is right over here in this particular plant. Now again, the picture given in your NCERT, it looks like this. Okay, it looks like this. This is again multiple carpels, but you know what? All the carpels are different. They are not fused together. They have individual ovaries. This is how it actually looks, and basically this is just inside part of it. Outside you will be having petals. This is champak. Though you might have heard about that flower, champak. It smells so good. Okay, so yes, this is known as miscellia. It's known as miscellia in scientific language, and yes, this is given in your NCERT. This is not something that I have made it up over here. So this is going to be miscellia. Cool. Done with this question also? If yes, give me a thumbs up. And by the way, Akash, good going, gave me the correct answer. Manoj, Macha, it's totally fine. You at least got to learn something, right? Let's go ahead with the next question then. Okay, perfect. Ayyo. One more question is there, sorry, MCQ, one more is there. Thank you, ma'am, most welcome. The correct sequence of embryogeny in dicots is? The correct sequence of embryogeny in dicot is what? Pro-embryo, globular embryo, heart-shaped embryo, or mature embryo? Globular embryo, pro-embryo, heart-shaped embryo, or mature embryo? Pro-embryo, heart-shaped embryo, globular embryo, mature embryo, or this? Manoj says the answer for this question is going to be A. What about others? Wanna try? The correct sequence of embryogeny in dicots. This is actually very relatable to our life. I'll tell you. So you have zygote. Zygote is a single cell thing. I hope you know that. The single cell divides. It is going to form pro-embryo. It is going to form pro-embryo. Okay. The pro-embryo which is there. Okay. When we are chotu people, you know, we eat a lot. We eat a lot and we become golu. So I'll just remember it as globular. Okay, we become globular. Then the globular grows up, then the globular grows up. We all have little crushes here and there. Okay, we have little crushes here and there as and when we grow, when we reach the teenage. So I can say it's a heart-shaped embryo. Heart-shaped embryo. Then we realize that the heartbreak is going to happen, and that's when finally we are going to become what? Mature. That's when we are going to have the mature zone out there. So this is how you can remember zygote, pro-embryo, globular embryo, heart-shaped embryo, and the mature embryo, which is why is the answer for this question A. Definitely it is going to be A. The zygote is not mentioned, but everything is being mentioned over here. Is this a good way to remember things? I hope it is. Okay, I know not apt for everyone, but still works. Can I go ahead? You wait a second, huh? Life is lost, huh? A is right. A is right. Yes, A is right. Perfect. So, guys, now finally we have completed all the MCQs, but I believe that all the MCQs that we have covered, with that you have got some additional information around all the 2 and 3 marker and 5 marker that we're going to discuss as well. So I hope it's not a waste of time. I hope you have learned something, right? Agreed or not agreed? Do you agree that, yes, ma'am, I have learned something? Or do you feel like, no, ma'am, nothing, I have not learned anything? Just talk about it and then we'll go ahead. Agreed. Okay, perfect. Guys, just give me a quick second. I'm getting a call from my mother. Just give me a... Okay. So sorry. Yes, I hope we're good to go over here. So 2-marker question. The first question is, write the significance of sporopollen. Now things are going to get a little more faster. Okay, so significance of sporopollen is what, Baba? Sporopollen is found where? In the outer layer, which is known as exine. And what's the use of having this? So let's read it out. Sporopollen is one of the most resistant organic substances forming the exine of the pollen grains. And you have been asked about the significance, so you have to mention them in points. So the first point that you have over here is, first, protecting the pollen grains from high temperature, strong chemicals, UV radiation, and the microbial attack. That is going to be the first point. The second point is going to be what? Because of its extreme durability, pollen grains with sporopollen get preserved as fossils, which helps the plant in survival as well. So this and this are the two significance that we have for sporopollen. And as I said, is the outermost layer, the exine is made up of sporopollen. I believe you do not have any doubt with that. Right? It's cool, right? Can I go ahead with the next question? Now, I'm not investing time for the questions that we have already invested time in. If it's okay, or do you want an explanation, I can definitely do that. Creative, I teach in Diksha here itself in the YouTube channel here. Okay. Perfect. Now let's go ahead with the next question. Give reasons. Micropyle remains as a small pore in the seed coat of the seed. What happens basically? Why does micropyle remains as a small pore in the seed coat of the seed? So if at all you remember, you had the ovule. The ovule had two integuments. Okay, the ovule had two integuments. One was outer and one was inner. So when I say fertilization is happening, the ovule is going to transform into what? It's going to transform into seed. What happens to these integuments? They are going to transform into the seed coat. They will be transforming into what? The seed coat. Okay. The outer seed coat which is there is known as testa, and the inner seed coat which is there is known as tegmen. Here also the opening remains as it's, and this is known as micropyle, Baba. And why do you think we need micropyle over here? We need micropyle for one thing. Papa, the seed is there. It will require some water. It will require some exchange of gases. Why exchange of gases? For respiration to happen, you need exchange of gases. Oxygen should be coming in and things like that. Why water to come in? Whenever water is there, that's when germination is going to happen. This particular seed which is there, it has the embryo. The embryo has to form the shoot and everything. No, for that you need water, because water which is going to come inside is going to activate some hormones that are going to help with the seed germination. Always understand that. That is exactly why, what do you have? You have the micropyle. So let's read it once. Okay, so micropyle remains as a small pore in the seed coat of a seed. Why? Because the micropyle of the ovule persists even after fertilization and becomes a tiny pore on the seed coat, because it helps in the entry of water and oxygen during seed germination. So it remains unchanged as a small opening. That's the basic logic. So here is the picture. I have this particular portion that you see over here is going to be the micropyle, and the point of attachment, if you see in some anatropous ovule, okay, it will be just near the micropyle. This is the hilum that is there. Anyway, the small opening which is there is the micropyle which is there. Cool? Clear? Understanding things or not understanding things? Okay? We're going ahead with the next part of the story. Apple and cashew are not called false fruits. Sorry, are not called true fruits. Why, Baba? Why apple and cashew are not called as true fruits? Because as I always said, true fruits is where you have the ovary. You have the ovary, and the ovary which is there, it is going to transform into a fruit. Only the ovary, nothing else. The ovule which is there, it will be transforming into a seed. Now sometimes you might have seen in some plants, okay, in some plants, over here you have the thallus, thalamus, and on the thalamus you have all the other parts. For example, you have the petals, and then you would be having the sepals and a lot of other things. So here also you have the ovary. Oh my God, here also you have the ovary. In this case, the thalamus, or probably this part that is known as pedicel, either of it can also take part in the formation of fruit along with the ovary. That is when you call them as false fruits. The question is, apple and cashew are not known as true fruits because it is not made by ovary alone, but there will be thalamus, or it could be pedicel, that could be helping in the formation of a fruit. Now, how do you frame this answer? How can I get your marks in there? Okay, you can frame the answer like this. First of all, you mentioned what is true fruits. True fruits develop only from the ovary after fertilization. Okay, now in apple and cashew, the edible part, or basically the fruit which is there, it develops from the floral parts other than the ovary, such as the thalamus and everything. Right? Other than the ovary it is happening. Now, like the thalamus in the case of apple, and pedicel, I told you what is pedicel over here. This portion that you have, the stalk-like structure that you have, Baba, it is going to be pedicel. Okay, I have to slow down. So sorry. So, yes, I'm just repeating it again. How do you put it down in the answer? First, you mentioned what is true fruits. You mentioned that true fruits only develop from the ovary. Done. Then you talk about the false fruit. What happens differently in false fruit? In apple and cashew, the floral parts, the other parts of the plant, they are also participating in the formation of a fruit. Okay, they are also going to participate in the formation of fruit. For example, in the case of apple, it is the thalamus. In the case of cashew, it could be pedicel. But anyway, they are participating. That is the reason. Now, hence they are called false fruits or accessory fruits and not true fruits. And now for you guys only. Okay, now for you guys only, I will be just showing you a picture. This is the picture. You know, you all have eaten apple. Okay, you all have eaten apple. You know, the edible part of the apple that you eat, the crunchy part that is outside, it is actually the thalamus. It is going to be the thalamus. So, ma'am, ovary is not taking part over here. Ovary is also there. But where exactly ovary is there? This portion, in the middle portion, that is the ovary. Surrounding the seed, no, you have the ovary, that is definitely there. But apart from it, other part is also participating. That is why I'm telling you. Okay? Cool. I hope this works. I hope this particular thing works for all of you. Hence, they are known as false fruit. Cool. This is very, very important. And by the way, for those of you who are wondering, what is this, ma'am? So they have mentioned mesocarp, endocarp, and exocarp and everything. Let me tell you one very important thing. The ovary which is there, it has some wall layers. This ovary which is there, when it becomes the fruit, it has some wall layers. I'll together call the wall layers as pericarp. They are all the ovary walls. The wall of the ovary. The pericarp can be later on divided into multiple types or multiple parts. For example, if I talk about mango. Just give me a second. Over here in mango, the thing that I peel off is a part of ovary only, the ovary wall only. What do I call this part as? You call this as exocarp. Okay, that is the thing that I peel off. The thing that I eat, that is also the part of the ovary wall, I call this as mesocarp. What do I call this? Mesocarp. And then there is an innermost region over here. Okay, there is going to be something like this over here covering your seed. So your seed is there. Okay, your seed is there with the seed coat and everything. But apart from it, you have one more thing outside it. This is going to be known as what? This one, which is going to be there, is known as endocarp. That is the hard shell around the seed. Okay, together these three layers form the pericarp, that is the outer wall of the ovary, or basically I can say the wall of the ovary. Now in some cases, for example, in the case of apple, what is happening? The ovary is right over here, it is inside. Okay, the ovary is going to be inside, which is why they have mentioned as mesocarp just around the seed, and then they have mentioned endocarp, there will be exocarp as well. Okay, so please do understand that. Is this question clear to all of you? If yes, can you give me a quick thumbs up? If yes, can all of you give me a quick thumbs up? If it's clear, if you have any doubt, you can ask me. Perfect. Let's go on to the next question out there. Differentiate between pericarp and perisperm. Can you differentiate between both of them? Pericarp, we just discussed about it right now. Pericarp is nothing but the covering. It is nothing but the covering of the ovary, the walls of the ovary, that is pericarp. What is perisperm? We discussed about perisperm also. Here, if you remember, ovary, inside the ovary ovule. Inside the ovule, sometimes the cells of the nucleus, they persist. Sometimes the cells of the nucleus, they persist. What do you call these cells as? Basically, I will call the cells together as perisperm. So pericarp is the covering of the ovary and perisperm is something different. And how do you frame the answer for this? Let's see. So pericarp is the wall of the ovary, develops into the wall of the fruit. Right, ovary turns into a fruit. That means the wall of the ovary will turn into the ovary of the wall of the fruit. Here you have three layers: exocarp, that is the outer layer; mesocarp, that is the middle layer; and endocarp, that is the inner layer. Now the mesocarp which is there, it can be fleshy. That means you can eat it up like that. Sometimes it is not. For example, in groundnut and all, you can just peel off the layers. There is no fleshy mesocarp and all. You can just peel off the outer thing that is there. So this is about pericarp. What is perisperm? Persistent nucleus is known as perisperm. Why it is there? It is there for the nourishment of the embryo. Sometimes, very rarely it persists, but if it's there, it's there. And please do remember the examples of perisperm. In few plants you find perisperm to be there. The best way to remember it is B square. Okay, you can remember this as B square. What is B square? Black pepper and beet. Both of them are going to start with B, so you can remember it as black pepper and beet. But basically both of them are examples of perisperm. Is this question clear to you? Do not forget, you can get questions related to this. Examples related to perisperm also could be asked. If this is clear, thumbs up. If you have any doubt, ask me. Okay, so we are going to go ahead with the next question, that is, how are parthenocarpic fruits produced by some plants and apomictic seeds produced by some others? So what do you mean by parthenocarpy? That is a new term. So parthenocarpy has something to do with the fruit. Okay, we know true fruit, false fruit. Apart from that, there are parthenocarpic fruit. What happens over here? So until now we have studied that the ovary is there. Okay, just a second. Ovary is there, ovule is there, ovule becomes the seed and ovary becomes the fruit. Right. What if I tell you that here fertilization is not happening? Fertilization is not happening. Still the ovary is transforming into a fruit. Do you think that's possible? Yes, that is possible. Ma'am, in that case, if at all fertilization is not happening over here, there will not be any seed. No seed will be there. No fertilization means no seed. Right? Right. These fruits which are going to be formed will not be having any seed. But ma'am, how is that possible? Listen to me. There are some hormones which are going to get activated, which is exactly why there will be ripening of the ovary and there will be formation of fruit. This is a natural phenomena in lot of the plants. Okay. Now sometimes we can also induce it. For example, I want seedless variety of fruit. I can induce some hormones into the plant such that the ovary will transform into a fruit without the formation of a seed. So if you have seen, the actual example for this is banana. Okay, this is banana. In general, the banana that we eat does not have any seed. You might have seen small dot-like structure. They are seed remnants. They are not seed, they are seed remnants. But in actual case, if at all, without parthenocarpy, if you see the banana can have seeds like this. They can have seeds like this. So as of now, we have cultivated plants to be like this, that now they are producing seedless varieties of things right there. We are going to show asexual reproduction. They are not going to show sexual reproduction out there. That is what I said. No fertilization is going to happen. So there are banana which can be producing like this. This is the form of this is the form of sexual reproduction where they have seed and all. But generally nowadays the plants that we see, all the banana plants that we see generally produced by asexual mode, and this is an example of parthenocarpy where they do not have seed at all. Always remember it. So how are parthenocarpic fruits produced? They are basically fruit produced without fertilization. How are apomictic seeds produced? They are basically seeds produced without fertilization. So here it is fruit, there it is seed. Okay, and and you have to explain that. How do you explain that? You can put it like this: parthenocarpic fruits produced without fertilization of the ovule. There is no fertilization of the ovule, right? There is no fertilization of the ovule. Second thing, the ovary develops directly into a fruit. Does it develop directly into a fruit? Yes. Okay. This develops directly into fruit. Can occur naturally or be induced by applying auxins and gibberellin? Yes, it can occur naturally, but we can induce it by giving hormones as well. The fruits are seedless as fertilization does not occur. Already mentioned about that. That is all about parthenocarpic fruits. Now here, apomictic seeds produced through apomixis, that is seed formation without meiosis and fertilization. Is it produced through apomixis? Yes, and basically over here seed without meiosis is happening, and without fertilization can we produce it? Yes. The embryo develops from the diploid egg cell or diploid nuclear integumentary cell. Either of these cases can happen, right? In the case of apomixis, either the diploid egg cell directly forms the zygote and things like that, or it can be the nuclear cell that directly forms it. That is true. Since there is no fertilization, the offerings are genetically going to be similar to the parent plant. That is also true. Seeds are formed without sexual reproduction. That is also true. So in both the cases, there is no sexual reproduction that is gonna be seen. Yakash ma'am, class will be winded up by 8:30. Okay? Now, I hope that question is clear, children. That question is clear. Will you be able to answer it? It's a 2-mark question. I thought I should be just discussing it, so I just did. If it's clear, can you give me a thumbs up? If it's clear, can you quickly give me a thumbs up? Yes, perfect. Let's go ahead. Differentiate between chasmogamous and clistogamous flowers. So how do you differentiate is a thing that we already studied. Chasmogamous are going to be the open one and clistogamous are going to be the closed one. But here, let's talk more about it. Flowers open at maturity, exposing the reproductive organs. That is chasmogamous. Clistogamous flowers remain closed throughout the life. Perfect. Next time, chasmogamous cross-pollination and self-pollination are both possible. Yes, they open flowers from here. Basically, cross can happen between different plants or the same plant itself, or basically self-pollination can also happen. In clistogamous, only self-pollination, autogamy is going to occur. Okay. Next point, require external pollinating agents such as wind, water or insect for cross-pollination. Obviously, if they are open, you will require so many things. But since they are closed, they do not require any pollinating agents out there. Next point, may fail to produce seeds if pollination does not occur. Yes, if pollination is not happening, definitely seed will not be produced over here unless and until they are apomictic and things like that. Clistogamous flowers, they always produce seeds. Why? Because they are very close to each other. They are closed, Baba. There is no option. Seed setting is for sure. Next, example is Viola oxalis commelina, the open flowers of them. Here also Viola oxalis commelina, the closed flowers of them. Even if you do not write the last point, it will be still fine. You can just write these things that we have already studied. Okay, so yes. Devaraj, ma'am, slowly. I'm so sorry, dear. The reason I am talking like this is because we had already covered this while we spoke in the MCQs. That's the reason. I'm so sorry. I know you could be someone who have newly joined over here. Okay, children, understood this topic? Do you need me to repeat, or is it fine? Or is it fine? Yes, perfect. Okay, the next question, distinguish between albuminous and non-albuminous. Again, something that we had discussed before. What do you mean by albuminous? Albuminous means endosperm is going to be there. Non-albuminous means endosperm is not going to be persisting back. Have we discussed it before? Yes. So how do we write it? Albuminous seeds, they are going to retain some endosperm even after seed maturation. Non-albuminous, endosperm is completely used up during the embryo development itself. Now over here, where do you like in albuminous seeds, Baba, who provides nutrition? It's basically provides nutrition to the embryo from the stored endosperm itself. The endosperm itself is the one that is going to provide nutrition, and along with endosperm, it can be cotyledon also. For example, monocots, endosperm as well as cotyledon could be there. In this case, anyway, there is no endosperm that is left behind, which is why the only source of nutrition is going to be what, Baba? Cotyledons. Do you have any other options? No, just cotyledons are going to be there. Got it? The next question, sorry, next point which is there is the example. Albuminous seeds, almost all the monocots, you can put it over here, wheat, maize, barley. Castor, as I said, is a dicot, but it's an exception which comes under albuminous. Over here, you have all the dicots out there, bean, pea, groundnut, all of it coming there. Clear? Already discussed it before. If it's clear, let's go ahead. If it's clear, shall we go ahead? Okay. Cool. So let's go. Draw a label diagram of mature two-celled pollen grains. It's clear. Fine. Thank you. Draw a label diagram of mature two-cell pollen grain. Dear, we have already made this diagram, I think, in the first part of this particular chapter itself. Do you want me to draw it again, or can we just skip this question? By the way, this question is important. Okay? Do we have to repeat it, or can I skip it? If you want me to draw the pollen grain again, I can do that. So just tell me repeat or not. Ma'am, skip. Okay, one person says skip. I have done it on the initial part of the chapter itself. I drew it, but okay, then we will skip. Guys, for those of you looking for the answer for this particular question, please move on for the MCQs. There we have spoken about it. Now, in the 3-marker question session, over here, we have the first one that we had already made, the label diagram of LS of the embryo of the grass. I hope this is clear to all of you. Very, very important picture. It could be coming in a 3-marker question. Do not miss it out at all. Okay. Now draw a neatly labeled diagram of a typical anatropous ovule. This is also been done. We previously spoke about it. That's why I'm telling you. By the time we reach over here solving the MCQs, we might have done a lot of questions out there. Right. So typical anatropous ovule is also been done. Do you want me to repeat it, or is it okay? Can I go forward? Can I go forward? So anatropous ovule, where did we do it? We did somewhere. Oh, I'm not able to find it, but yes, you will be able to find it because you will get the PDF for this, right? Okay, anyways, I'm not wasting time. Skip and continue. Okay, fine, cool. Let's go ahead then. Apomictic seeds, this question we have done. Why are some seeds referred to as apomictic seeds? Why, Baba? Because... Move on, ma'am. Okay, this also we have done. Everything we have done. Now we entered into the 5-marker questions. Let's go. This is something that we had not done. So plan an experiment and prepare the flow chart of the steps that you would follow to ensure that seeds are formed only from the desired sets of pollen grain. Name the type of experiment that you carried out and write the importance of such experiments. Important question. Listen to me. What is a question that has been asked? You have to plan an experiment where you are going to make sure that the seeds are formed only from the desired sets of pollen grains. Only from the desired set of pollen grains. That means you're talking about which process. Name the experiment that is being carried out. Any idea? It is known as artificial hybridization. In artificial hybridization, or you also call this as controlled pollination, what are you going to do? You're going to pick the plant. I like this plant because it is drought resistant. I like this plant because it is pest resistant. And I am going to make sure that I transfer the pollen grains myself and make the fertilization process and everything go smooth. Okay, here it needs human intervention, and that is exactly why I'll get the desired set of pollen grains to be transferred to form the seed that I want. And how is this process going to take place? Let's talk about it. The first thing that you would need is basically, suppose this is a flower that you have. In the flower, you have the female and the male part. Just think like that. If you have the female and the male part, what do you call this type of flowers as? I will call this as bisexual flower. Okay, I am going to call this as bisexual flower. It has both the male and the female part, right? Done, ma'am. Now, in this case, there are chances that the pollen grains from this part can land on the stigma on this particular plant itself. That means self-pollination can happen. Do I want that? No. I want some other pollen grains of the plant that I am looking forward to come over here. So what do? I cut this off. I cut the male portion off. What do you call this as? The first thing, emasculation. I will call this as emasculation. emasculation. You can remember the masculine part is removed, that is why you call this as emasculation. Okay, done. Now, by the way, this is a bisexual flower, that is why you need to do this. If at all it is not a bisexual flower, it is a unisexual flower, you do not need to do this. Okay, Manoj says emasculation, bagging, dusting, pollen grains and rebagging. Perfect. Now, after this, you have got the flower. This is your flower, which is having the female part. You do not want any other male parts to come over here, basically pollen grains to come and land over here, which is why what you're going to do? You are going to do bagging. You are going to do the next process, which is known as bagging. Now, while you bag, you want to prevent contamination. Apart from that, you also want to do one more thing. You want to make sure that the stigma is maturing to become receptive. Okay. Now, once that has been done, what I'm going to do is, the other plant that I had, I'm going to take the pollen grains from that particular plant. I will be collecting the pollen grains. Collect the pollen grains. Now, after I collect it, what do? I take that and I come over here. I remove this bag. I am going to remove this particular bag and I'm going to put that pollen grains over here. I am going to dust the pollen grains. I am going to dust the pollen grains. So, after third one, collection of pollen grains, I am going to dust the pollen grains myself. And after dusting, what will I do, Baba? I am going to put the bag again. Why do I put the bag again? Because I do not want the contamination to happen. Now also some pollen grains can come and land. Do you know that? Multiple pollen grains can come and land. Multiples of them can form pollen tube. Only the one can reach ahead. That's the thing. So it can happen. I want to prevent that. That's why I cover it up. This is known as rebagging. This is known as rebagging. And finally, I do one more thing. I tag it up that this is the plant and this is the plant that I crossed it with, so that I have an understanding that what I'm going to get in the future. What type of hybrid plant do I get in the future? So this is how you have to go about. Name the type of the experiment. It is known as artificial hybridization or controlled pollination. Why controlled? Because you are doing in a controlled setup. Okay, you are the one who is choosing all of it and doing it. Now, write the importance of such experiments. What is the importance? Suppose I want some hybrid characters to be seen in the plant. I want a typical type of plant to be formed. Then I'll take that up. And obviously, the good part of the story, the next good part is that I am going to get varieties of plant that are going to be drought resistant, pest resistant, and it is going to be helping in its survival. The plant can survive even better in that particular case. So I can talk about it, but how do you frame the answer? You're going to frame the answer like this. This is a 5-mark question. Every answer that you write in your examination have to be written in points. Please write them in points. Aim to ensure that ovules of selected plant parts are fertilized only by desired pollen grains. Steps: select parents first, then do emasculation, then do bagging, then pollen collection, then artificial pollination, then rebagging and tagging. And for the flowers which just has unisexual flower, it's not having the male part, you do not need to do emasculation. This is the first part of the question. Second part, name the type of the experiment. It is artificial or controlled pollination. Write the importance. They prevent contamination by unwanted pollen and help in producing hybrids with desired characters such as higher yield, disease resistant, better quality, etc., which is good for crop improvement program. Is this question clear to you? If it's clear, you will be getting the 5 marks straight. If it's clear, let's go ahead. Perfect. Cool. Manoj, for Manoj it's clear. What about others? Champs? Akash, Baba, clear? Okay, cool. Mahantesh is also there. Hello. Okay. Picture related to pollination is given alongside. Answer the questions that follow. Will this pollination confirm fertilization? This is the pollination. Insect is there and the flower is there. Okay. And you have been asked that basically, will this pollination confirm fertilization? What do you think is the answer for this question? Listen to me. Any pollination which is there, pollen grains have landing over here. Okay, this is the basic logic that you need to have in your head. You have this stigma right over here. Where is it? Come on, work. Okay, you have the stigma right over here. Pollen grain can land over here. They can form pollen tube, multiples of them, but there is no guarantee that yes, it will be going and fertilizing. It will be helping in the transfer of gametes. There is no guarantee, never ever. So will this pollination confirm fertilization? The answer is no. Justify your answer with suitable reasons. The reason is that pollination is going to be helping in the transfer of pollen grains. Pollination is going to help in the transfer of pollen grains. Right? it does not guarantee the fusion of pollen grains, or sorry, that fusion of the gametes out there, then how can you say? So how will you frame the answer? You can frame the answer like this. More pollination does not ensure fertilization. Pollination only brings pollen grains to the stigma. It is just a transfer process. Next thing, fertilization occurs only if the pollen is compatible. If it's compatible, if it's germinate, after germinating it has to go form the pollen tube and basically fuse with the gametes. So that is the whole process of fertilization. Finally, if the pollen grains is incompatible, it will not germinate and fertilization will also not happen. So pollination has taken place, but it's not compatible. Do you think it will form the pollen tube? No. Do you think it will go and fuse? No. And even if it's compatible, we never know that whether it will be doing the entire process or not. So always remember that. Okay, Ma'am Broha, she's teaching us. Okay, so someone asked me about dinner. No, I didn't have dinner. I actually didn't have lunch also. I didn't have breakfast as well. I'm surviving on, I think, one paperboard juice which I'm surviving on Good Day biscuits. Yes. Now, the next question that we have right over here is, what are the floral rewards provided by the plants to the insects to revisit? For example, I am an insect, okay, and I come to the plant. Okay, I come to the plant. What do you think that will make me come back to this plant again and again and again? Obviously, plant cannot speak and say that, Macha, be here, be my friend. So there is only one way, or there are a few ways out there. The first way would be basically production of nectar. The animals, the insects, they like it, so they will come to drink the nectar and all. Second way could be pollen grains itself. That Macha, you take some pollen grains, but while you take some pollen grains, I am pretty sure that on your body some pollen grains will be there and that is going to be transferred. So plants are okay with giving some pollen grains to eat also. So they do that. So definitely they do that. The third thing is shelter. Some plants are there, you know, you might have heard about yucca and the moth. Yucca is a plant and moth is a moth, an insect. The yucca plant provides the moth the safe space to lay their eggs. So that is also a way that is also a way where you can help in the process of animals or basically insects to come and revisit. Okay, so B part is also done. Ma'am, you come directly to my home, my dinner is ready. Oh, so sweet of you. I wish I could come. Okay, anyways, then the last question is, mention the characteristics of flower in which pollination other than the mention the characteristics of flower in this pollination other than the floral rewards. Can you tell me, guys who are watching this video, can you tell me what are the other features of the flower that is going to be pollinated by insects? Other features apart from the floral rewards? Any guesses? Any guesses? The pollen should be big, something like that. Is it like that? Happy Guru Purnima, ma'am. Is it? Thank you. Is it Guru Purnima today? I'm so sorry. But thank you. Guys, I'm waiting. Come on. Tell me. Characteristics of flower. Okay, pollen grain should be sticky. Definitely, pollen grain should be sticky, otherwise it is not going to get transferred. So that is the first thing that you need foremost. The next thing should be what? The flowers which are there, they have to be colorful. Okay, the flower, they have to be colorful, they have to be large. The next thing that is there is what? If there are so many flowers, they should be coming together to form inflorescence. If they are small flowers, they should be coming together to form inflorescence such that they look big, okay, such that they look large, and basically insects can come easily, track them and come and do the process of pollination. The last one is that if at all there is a flower that is pollinated by any bee or beetles, they have to be producing some foul smell. They have to produce foul smell for attracting them. So these are the few things which are going to be there. So what do you say? Mention the characteristics of flower in this pollination other than the floral rewards. Large, colorful and fragrant flowers to attract insects. Second thing, sticky or spiny pollen grains so that they can attach to the insect's body. Third thing, stigma is sticky to capture the pollen brought by insects. And lastly, the foul smelling flowers in the plant pollinated by flies and the beetles. Is this clear to all of you? And children, I'm telling you again, if you want all the notes, go and click on the link that is given there. That's an all-in-one page. You will find all the YouTube videos tagged up there, subject wise. It will be very easy for you. Okay, and there is no price. There is buy now, but actually it's for 0 rupees. There is no price for it. Also, please join our WhatsApp channel. Okay, perfect. So Mahantesh said it's okay, he has understood. So I'll go ahead, and here is a question. And this question is actually a tricky question. I want all of you to pay attention and solve it. Complete the given tabular column with respect to the male and the female gametophyte of angiosperms for the mentioned characters. Okay, which advantage offered? Okay, that is the next part. So first we are going to solve this one. This is the mature male gametophyte, this is the mature female gametophyte, and you have to, okay, you said that you will send the link, ma'am. Okay, so Manoj, please wait for the class to end. Once the class ends, put down the comment in the comment section. I'll come and reply back to it with the link. Okay, I'll request some time to go back. And the second chapter, sadly, I do not feel that I would be able to complete it today because we are still stuck in the first one. But if it's okay for you, I have done the second chapter's one shot with all the questions that I was planning to discuss over here apart from the MCQs. You can go ahead and watch it if it's okay. Okay, and I really didn't know that you had an exam tomorrow because I was expecting that for all of you, it will be happening towards Monday and things like that. So I'm literally very sorry. Okay, so over here, the question comes back. Number of cells present in the male gametophyte. Can you tell me, guys, the male gametophyte, what are the number of cells present? I need only the important questions. Okay. In that fixed question video, it's there. I'll send you the link, dear. Please make sure you put down a comment in the comment section once this particular whole live ends. Okay? Not now, in the comment section. Okay, guys. Number of cells present in the male gametophyte. What are the number of cells present in the male gametophyte? Any idea? Male gametophyte means you're talking about what? Pollen grain. In pollen grains, how many cells are there? In pollen grain, how many cells will be there? You have two cells, right? Generative and vegetative. Generative and vegetative. Now again, if generative cell is going to divide, what happens? It is going to form two other gametes. It is going to form two other gametes. So always remember, in total, it can be either two or basically it can be three. Children, are you paying attention, or am I the only one who is solving this question? Can you try for the female gametophyte, that is the embryo sac? How many cells are present in the female gametophyte? Generative and vegetative. Manoj has now said the answer. Perfect. Okay. Apart from it, always remember the generative cell can divide into two gametes also. No? So you always write two, number of cells can be either two or it can be three. Okay. Now here in the embryo sac, how many cells do you have? How many cells do you have in the embryo sac? Come on, come on, come on. Only one. In embryo sac, seven cells are there, right? We have 7 cells, 3 antipodals and 3 synergids and 1 central cell. Akash, please have food, dear, please. Okay, you will be having 7 cells in the embryo sac. In the male gametophyte, 2 or 3 cells. So over here, I'm going to bring about the condition like this. It will be easy for you to remember like this. So number of cells present in the mature male gametophyte would be 2. 2 as in 1 vegetative and 1 generative, or it can be 3, where 1 vegetative and 2 male gametes can be there. In this particular case, mature female gametophyte, embryo sac, there will be 7 cells. There will be 7 cells, 3 antipodal, 2 synergid and 1 egg cell. Now number of gametes present. Can you tell me the number of gametes which are present over here? Number of gametes which are present in the male gametophyte. Come on, fast. Number of gametes which are present in the male gametophyte. Two. Exactly. We have 2 gametes over here in the male gametophyte, 2 gametes. What about the female gametophyte? Mahantesh also said 2, Manoj also said 2. Perfect. Now Baba, in female gametophyte, how many do you have? How many gametes do you have? 1. Only one female egg cell is there, right? So 1 would be the answer for that. Cool. Now the next question is, type of cell division involved in the development of mature male gametophyte. What type of division do you see in the development of the mature male gametophyte? Any idea? In the formation of pollen grain, what type of division do you see? what type of division do you see? Is it mitosis? Is it meiosis? It is going to be meiosis, is it? That is for the formation of microspores, right? You have got the microspore. The microspore is going to divide by mitosis to form the pollen grain, right? It is going to do the mitosis process. Remember, small thing, first meiosis, you get microspores. Perfect. I'm not disagreeing to that. But post that, it is going to be mitosis, no? Microspore dividing into two cells, vegetative and generative. Remember, mitosis. So it's going to be mitosis, Baba. It is going to be mitosis. Now, what about the embryo sac? What is the division that happens for the formation of embryo sac? So Mahantesh gave me the correct answer, mitosis. Aesthetic is there. Hello, dear. Oh, I thought microspore. It's okay. It's okay. At least now we have got this type of questions could come, right? We have got an understanding. What about the female gametophyte? Female gametophyte also, it is going to be mitosis. Okay, mitosis only. Perfect. 3 rounds of mitosis happens over here for the formation of female gametophyte. If you remember, free nuclear division and all, right? So 3 rounds of mitosis is going to happen there. Cool. Now, the ploidy of the cells. Can you tell me the ploidy of the cells? So here also mitosis, there also mitosis. Now I am asking you, what is the ploidy of the cell? Over here, in the male gametophyte, the generative and the vegetative cell is there. What's their ploidy? Are they haploid? Are they diploid? Are they triploid? What is their case? In this particular case, in the ploidy of the cells of the male gametophyte, haploid, ma'am. All of them are going to be haploid? Yes. Why? Because it formed from the microspores and they were haploid. So obviously all of them are going to be haploid. What about this case? What about the female gametophyte? What about the female gametophyte? Manoj says haploid or diploid. Manoj, you're talking about this guy, the male gametophyte. Can it be haploid as well as diploid? Do you think so? Okay, Mahantesh says haploid. Guys, I'm asking you fresh, okay, fresh options. You might have to give the answer once more. It's because you were saying haploid. Now, from this point onwards, I am asking about the female gametophyte. The cells of the female gametophyte, are they haploid or they diploid? Aesthetic says haploid, okay. What about others? All the cells are haploid? And this is no fertilization. Do not include fertilization over here. Plain embryo sac without fertilization. Mahantesh says haploid. So Baba, if at all you remember, in the embryo sac, what all do you have? You have the antipodals, haploid, agreed. Synergids, haploid. Egg cell, haploid. What about the central cell? Central cell has 2 nuclei, 2 polar nuclei. It is going to be diploid. So you have to mention like that all cells except central cell is going to be haploid. Got it? You have to mention like that. This is a tricky question. If you make it wrong, you might lose your marks. This is actually a 5-marker question. So from a 5-marker question, the question came in 2 parts. This was the first part. So 3 marks comes from here. Got it? Did you get it, children? Do you have doubt? Please ask me. If you have any doubt that is there in your head and you're really confused about it, please ask me. No. So they gave me thumbs up. So I believe it's all cool. Aesthetic expressor here, it's clear. Akash, Baba, clear? And Sahil, dear, I would require some time to read it. And by the way, I cannot, predict the cutoffs or probably things out there. So it will be really good if at all you do not rely on me for the cutoff thing. Okay? But I could really help you towards the end. Akash, okay, fine. Let's go ahead. Now, there's the other part of this particular question which says, which advantage offered by seeds help angiosperms to adapt to changing environmental conditions? So basically, they're asking you, what are the good features of the seeds? What are the good features of the seed, Baba? Any guesses? So seed which is there. Seed is a unique feature of what basically you had gotten it after you have, algae, then bryophytes, then pteridophytes, then gymnosperms and angiosperms. From the point you have started getting basically the gymnosperms and angiosperms, that's when the seed came into the picture. Until then, for everything you were relying on water. You were relying on water for pollination to happen, for this to happen, for that to happen. Now since seeds have come, you do not rely on water anymore for fertilization and anything. Rather you require pollinating agents. It can be wind, water, insect, anything out there. So that is the first good thing about it, that even without water this can survive. Second thing, yes, dormancy is one more thing. For example, I will say that seeds which are there, they are really clever enough to remain dormant. They have all the reserve. Okay, they have either cotyledon, they would be having either endosperm to make sure that the embryo which is formed, it is right, very cool, it does not have any problem, it just stays there. And it is really good because until and unless it gets a favorable environment, it will not go further and germinate because it has a reserve food and it's okay with that. So that is one good feature about seed, that seed is made in a way that it will be having embryo along with some reserve food. It can be either endosperm or it can be cotyledon. Okay, that is the next point. The third point that could be there is basically seed is very important in terms of what is there inside, that is the embryo. Again, to provide nutrition and all, I told you that cotyledon, endosperm and all is there. Apart from it, seed has a seed coat which covers the embryo really well, such that it does not have any damage. Now, if it does not have any damage, obviously this maintains or makes sure that the embryo will develop in the future whenever it is required. Got it? These are the few points about seed. We'll just read about it. First is protection and dormancy. The seed coat protects the embryo and allows it to remain dormant during unfavorable condition. Very important. If at all the embryo is not safe, do you think that the plant is going to survive later on? No, Baba, it's not going to be born there. Food for the embryo. Seeds have stored food that nourishes the young plant during germination. All the plants are very important for us. If at all they die off, we will become extinct later on, right? So seeds are also super important because they are making sure that the embryo is safe. Third one, dispersal. Seeds are adapted for dispersal by wind, water or animals, helping plants spread to new areas. Now, seeds which are usually there are small, they can be easily dispersed, and while I say they are dispersed, again, we are making sure that their hierarchy is being continued. They are not stopping anywhere up there. Got it? So these are the few features that are of advantages by the seeds that are offered to the angiosperms, which helps in surviving changing environmental conditions or basically unfavorable conditions out there. Cool? Does it work? Have you understood it? Or do you need an explanation? Cool? If it's cool, if you have understood it, can you give me a thumbs up? Okay. Ma'am, I can't attend part 2 as I have math exam. Oh my God. It's okay, Manoj. I'm truly very happy that at least you came and said that and you're concerned about it. And this is so sweet of you. Okay, guys. Let's go ahead with the next question. Explain the different, let me check how many questions are there. More. Okay, soon gonna end. Cool. Explain the different outbreeding devices developed by the flowering plants. What do you mean by outbreeding devices? 3 more questions are there. Please bear with me, okay? Outbreeding devices means basically you do not want self-pollination to happen. You want cross-pollination or some other pollination to happen, but not self-pollination. Which is why the plants have come up with ideas that, okay, if this happens, obviously we can prevent self-pollination at least to some extent. These mechanisms which are there to prevent self-pollination and to promote cross-pollination is known as the outbreeding devices. Okay, it is to prevent the inbreeding depression. It is to prevent the inbreeding depression. Okay, ma'am, are you covering genetics in the next class? Yes, Mahantesh, I'll be covering genetics in the next class. Okay, so that is on Friday. Okay, so here in this inbreeding depression, why do you call inbreeding depression? Because suppose I am a plant and have all the toxic traits. I'm not good at anything that is there, which is very much important for me to survive. Whatever progeny that I'm going to form, it's going to have the same thing. It's basically getting depressed because of the same things out there. It will not be able to survive. That is what inbreeding depression is. To prevent that, you need outbreeding devices. The first device that we have is time difference. Easy way to say. Time difference is basically when pollen release is there, but the stigma which is there is not receptive. It is not receptive. Pollen release and the stigma receptivity is not synchronized. That is the first thing out there. Very important. Second way, spatial separation. For example, over here you had the stigma right at here and the anther and everything right over here. Do you think it will be easy for pollination to happen? No. This is spatial separation. Anther and the stigma are positioned differently. Now obviously there are so many complicated terms to it. You can, if at all you want, you can remember like that, but I will not suggest it because this will be more than enough. Okay, in your NCERT it is not given, this is dichogamy, there is this gamy, there is that gamy. I really do not want you guys to suffer with all those terms. That is why I am telling you. You can just remember this. It would be more than enough. Cool? Next thing, self-incompatibility. Now this is a genetic mechanism where what is happening? Genetic mechanism as in, for example, the pollen grain landed up over here, but somehow, you know, for every interaction, for every pollen grain that comes over here, there is some chemical interactions that are happening up over here. If it is good, if it is compatible, then only it will allow the formation of pollen tube. But right over here, what is happening? It's a genetic mechanism that is stopping from this process to happen. It does not allow self-pollination to happen or self-pollens to come and fertilize the ovule, does not allow the formation of the pollen tube itself. So until now we have studied about three things. One is time difference. Okay, both of them are getting mature at different times. Second thing is spatial separation. They are arranged differently such that no pollination can occur naturally. The third one is self-incompatibility, which is a genetic mechanism that does not allow self-pollination to happen, or basically the pollen grain to germinate on the stigma out there. The last one which is there is the production of unisexual flowers, and this is something that we had discussed before. What do you mean by production of unisexual flowers, and how does it prevent it? So the best example for this is dioecious plants. In case of dioecious plants, as I mentioned, there is one plant, there is another plant. Here one plant is going to be female, this plant is going to be male baba. Here in the female plant you will just have the female part, here in the male plant you will just have the male part. Okay, what is going to happen? Do you think self-pollination can happen? Arey Baba, it does not have the male part. How do you think self-pollination will happen? It is not going to happen. It is not at all going to happen. So what's the only thing that can happen? Xenogamy, that is true cross-pollination. This is dioecious type of plants. I'll tell you, they are unisexual plants. The flowers are unisexual, the plant is also unisexual. Got it? Here you will only see xenogamy. There is no autogamy. There is no geitonogamy. Questions can come on the basis of that. No xenogamy, no autogamy. Please remember it. Okay? Example for this is papaya. Now, under unisexual flowers, there is one more type of plant, monoecious plant. Monoecious plants means what is going to happen? They can be in two scenarios. Here one, where both the sexes are in one flower. This too, anyway, we do not want. This will promote self-pollination. We do not want that. Now, there is one more scenario where there is a flower with the female flower and there is a flower that is a male flower. At least you can prevent autogamy, right? At least you can prevent autogamy. Geitonogamy will not be prevented, but at least autogamy. And if the flower is alone like this, there are chances some other pollen grains will be coming and landing. That's a good part of it. So this is a monoecious plant. This is also a monoecious plant. Monoecious means they are going to be bisexual plant. The plant is bisexual. Do not mug it up. Understand. The plant is bisexual, but the flowers can be unisexual or bisexual. I'm talking about unisexual flowers in the monoecious plant. Here, to some extent, at least autogamy is for sure prevented, but I can also prevent, at least autogamy is prevented, and that's a big thing. Okay, geitonogamy and nothing else will be prevented, obviously, but in some ways it is helping in cross-pollination to happen. That's a good part of the story. Example for this is CM, Chief Minister, castor and maize. Castor and maize are the examples for this. Is it clear? These are the few things that are there. They are the outbreeding devices. If you have any doubt, ask me. Throw the doubt at me. Aesthetic, are you gonna do second chapter in the next class, ma'am? Yes, dear. I'm so sorry. I was actually planning to do this in this class itself, but apparently it couldn't be done. So next time, probably I'll be coming up with lesser MCQs. MCQs take a lot of time. Okay, but I still believe that it was useful for you in some ways, the class, right? Now, yeah, so this is the plant, this is the female plant, this is the male papaya plant here in castor. Okay, here in the case of castor, you would be having both male and female in different flowers, not on the same flower, different flowers. Anyways, now here is a question that is, okay, double fertilization is a unique event taking place in flowering plants. Briefly explain. So direct question on double fertilization. There is no too much to talk about it. Okay, there is no too much to talk about it. So I'll say what is there. What is double fertilization? In flowering plants, two male gametes enter the embryo sac, right? Two male gametes. One male gamete is going to fuse with the egg cell. What does it form? Zygote. What do you call this as? Syngamy. The second male gamete fuses with the two polar nuclei of the central cell to form the triploid primary endosperm nucleus. This is known as triple fission. Because two fusions occur in the same embryo sac, I'll call this as double fertilization. That is a basic line that you have to mention. If at all you can draw the diagram and you can explain that as well. Okay, diagram of the male gamete and the fusion and things like that. Just like how we were drawing until now. Cool. Okay, now there is one more part to this particular question and I want you guys to try it up. Arrange the following terms in correct developmental sequence: pollen grain, sporogenous tissue, microspore tetrad, pollen mother cell, and the male gamete. And the male gamete. Come on, you have to arrange them in the correct sequence. And by the way, everyone who is watching this video now and have not hit on the like or the subscribe button, make sure you do so. Any idea how do you arrange them, all of them? Arrange all of them in correct sequence. First, what do we have above? We would be having sporogenous tissue. Inside the sporogenous tissue, okay, this is sporogenous tissue. Inside the sporogenous tissue, what do you have? You would be having the pollen mother cell, or basically microspore mother cell. The microspore mother cell which is there, it is going to undergo division and it is going to form what? The microspore tetrad at the end. The microspore tetrad, they are going to dissociate from each other and are going to form what? The pollen grain. The pollen grain later on will be dividing to form the gametes, the male gamete out there. Yes, so pollen grain, someone said pollen grain first. No, Manoj, not pollen grain first. First is going to be sporogenous tissue, then all of it is going to start up. At the end, second last, you are going to get pollen grains. Okay, be careful. When you get questions like this, you might feel that it's very simple, but still put in your efforts. Okay, just check it once more and then give the answer. Got it, ma'am. Guys, is this question clear? I think there's an easy question out there. Shall I go ahead with the next question? Perfect. Cool. Now this question is very, very, very important. It's like the previous year question, a very, what do you say, question out there, and I am pretty sure you will be able to try it. Okay, double fertilization is a unique feature of angiosperms, and the products of this double fertilization are what? Zygote and primary endosperm nucleus. Do you agree? Yes, ma'am. Next thing, in this context, when a hexaploid plant is pollinated by a tetraploid plant, find out the ploidia, zygote and primary endosperm nucleus. That is the question. What is the question? If a hexaploid plant is pollinated by a tetraploid plant, that means tetraploid plant is going to be the male plant. This one is going to be male and this one is going to be female. Why do you say that? Because here it is written, it is pollinated by tetraploid. Now what do you mean by all of it? You have been asked what is the ploidy of zygote and primary endosperm nucleus. That is a simple question that is being asked. First of all, hexaploid means 6n, tetra means 4n. Hexa means 6n, tetra means 4n. Now we have studied that basically when you talk about the gametes, or any other cell basically which is there, it will be having, ma'am, is it a type of HOTS question? Yes, definitely. Yes, it is. Okay, so over here what is going to happen, when you form the gametes, you want to make it half. Okay, you will not directly make it haploid, rather you will make it half. If it was diploid, you will make it haploid. Here if it is 6n, I will want to make it 3n. That is the rule. Normal cell will be like this, but the cells, the gamete, for example, when you talk about the egg cell over here, or probably the synergid over here, or probably the antipodal over here, okay, all of them, they are going to be having half the number of chromosome. If this is a normal number of chromosome, half of it is going to be what? So I am just writing over here, half of 6n is going to be what? It is going to be 3n. So this is going to be 3n in nature. This is going to be 3n in nature. Antipodal is also going to be 3n in nature. What about the central cell? Central cell, if you remember, it has two polar nuclei. It has two polar nuclei. One nuclei will be 3n. Other nuclei will also be 3n. right? One nuclei is 3n, other nuclei is also 3n, which is why it is going to be just like this. Just like how we had normally also. If we started with a central cell, it was diploid. Normal cells were also diploid. So central cell will have the same ploidy as the plant, which is there. Got it? Until this particular portion, is it clear to you? If it's clear, can you just give me a thumbs up? Hello, Mirage. Okay, guys, until this particular point, what I'm saying, if it's clear to you, can you give me a thumbs up? If you have any doubt, you can ask me as well. Okay, clear, perfect. Now, when you talk about tetraploid plant, that is a male part. Again, here you just have to form the male gamete. Here, you just have to form the male gamete. In the male gamete, again, you will just do half of it. Half of it, gamete is going to have half of it. So 4n, half of 4n is what, Baba? 2n. It will become 2n. Now, what do you want to form? You want to form the zygote. What is a zygote made up of? Zygote, the first thing that is made up of female plus the male gamete. Okay, so I am going to make it up. Listen to me. Female gamete is having 3n number of chromosome. Male gamete is having 2n number of chromosome. What is a ploidy of the zygote that is being formed? It is going to be what? It is going to be what? 3n plus 2n. It forms what? Ma'am, show diagram of angiosperma. Angiosperm is basically flowering plants, dear. Okay, it becomes 5n. It is going to become 5n. This is the ploidy of zygote. We have got it. It is going to be pentaploid. Penta means 5. Pentaploid. Okay, now I have a question. The next one, primary endosperm nucleus. How it is formed? Primary endosperm nucleus is being formed by the fusion of central cell. Okay, central cell's nuclei out there, plus the male gamete nuclei. Male gamete nuclei. Central cell nuclei, what is the ploidy? We have already found it is 6n. What is the ploidy of the male gamete? It is 2n. What will be your answer for this, Baba? What will be the answer for this? Any guesses? Akka, angiosperm diagram, please show. Angiosperm is a flowering plant, dear. You just have to make the diagram of the flower. I'll be showing you towards the end. Don't worry, okay? Just wait for some time. So, okay. Manoj says 8n is the answer. Yes, 6 plus 2, it is going to be 8n. Octaploid. It is going to be octaploid. Yes, aesthetics also gave me answer. Children, I hope in that case you have got it. Right? Got it, right? But I'll still request you, try to solve this question once more. Okay, try to solve this question once more. It is definitely going to help you out. Perfect. And do it by yourself. Don't take any help. Watch this video also. Here I have given everything which is required for you to write the answer. Make sure you look at it as well. Okay. The next thing that is there is, we are doing the last question. Do not be worried. Okay, enough for today, ma'am. Okay. Double fertilization is reported in plants of both castor and groundnut. However, mature seed of groundnut is non-albuminous and that of castor is albuminous. Explain the post-fertilization event responsible for this. Explain the post-fertilization event responsible for this. So what do you have? Double fertilization happening in both castor as well as groundnut. One is albuminous, other one is not. So how do you explain? The one that is albuminous will be having some endosperm that is left behind. It is not going to form the cotyledons by total difference of it. Okay. The next one which is there, in the case of albuminous which is there, they will be utilizing, non-albuminous which is there, they will be not utilizing the entire endosperm. Endosperm will be left behind. That is there. Sorry, I'm so sorry. I'm distracted. I'm so sorry. Okay, got it. Fine. Now listen to me very carefully. I'm talking about castor and groundnut. The question is, one is albuminous, another one is non-albuminous. How do you explain what is different in both of their post-fertilization event? That means after fertilization, what is going to happen? That is the question. So in albuminous, in the case of albuminous one, the endosperm which is there, it is persisting. Okay, it is persisting. That is the difference which is there. And along with that, the cotyledon is being made. In the case of non-albuminous, no endosperm is going to persist back, the endosperm is completely utilized by the embryo there for the formation of cotyledon. That is what you have to write. So here is the answer. In both castor and groundnut, double fertilization occurs, forming the zygote, which develops into embryo, and the primary endosperm nucleus, which develops into the endosperm later on. In castor, the endosperm persists in the mature seed, hence the seed is albuminous. Whereas in the groundnut, the endosperm is completely consumed for the formation of what? For the formation of cotyledons, hence the mature seed is non-albuminous. This is the change that you are going to have. Okay? Thank you so much, Shimon. His Weber, ma'am, for us this chapter is not there, is it? Okay, Shimon Joseph sir is teaching us everything. That's really good. Okay, now going ahead with the next part that is there. Banana is a parthenocarpic fruit, whereas oranges show polyembryony. How are they different from each other with respect to seeds? Can you guess, Baba? Banana is parthenocarpic. Parthenocarpic means what? Parthenocarpy. Banana, no seed. Banana means no seed. No seed. Without fertilization, you are forming the fruit. That is known as parthenocarpy. What is polyembryony? Within the seed which is being formed over here, by the way, the seeds are being formed with the help of apomixis, okay, and within the seed you have multiple embryos being formed. Here also there is no fertilization, but at least there is formation of seed, and in the seed there are so many embryos as well. In the case of parthenocarpy, there is no seed, only fruit is being formed out there. Okay, got it? So how do you write that answer? In the case of banana, the ovary develops into fruit without fertilization. The reason I'm not explaining it further is because we had discussed it before, right? The ovary develops into fruit without fertilization under the influence of plant hormones. Hence, the fruits are seedless. On the other hand, orange, which is there, more than one embryo is formed in a single seed. The additional embryos arise from the cells of the nucellus. So I spoke about it. If this is the ovule, if this is the ovule, inside the ovule you have the cells of the nucellus, and within this you would be having the embryo sac. Within the embryo sac, these cells are going to get into and make multiple embryos. That is basically first apomixis. Why? Because the cells are getting inside and forming embryos without fertilization. That is apomixis, and where multiple embryos are being formed, that is polyembryony. Okay? Got it? Works? Cool, right? Done? Everything is super clear. Everything is done. So that's all. My dear children, ma'am, link. Yes, I'm giving you link. Thank you, ma'am. You're most welcome, aesthetic. Manoj, as I told you, this live is going to end. Okay, I'm going to end this live very soon. What you can do is, once the live is ended, comment down. Not here, comment down, because there only I can. This live chat will go when the live ends. That's why I'm telling you to comment down. Okay, I'll give you the link there. Cool. So that's all, and thank you so much for being there. I really want to tell you one thing, that make sure you go check out the AIO page. The link is there, okay, in the description section of this particular video. Very, very important, because this is where you will get everything, biology, physics, mathematics, everything for free. And all of the chat, whatever has been happening in YouTube, it's been arranged properly, and you are going to get the notes as well. So check it out. That's all. So that's all. Lots of love. Bye-bye. Please take care of yourselves. Bye.